All right. Hello, everyone, and welcome back to my channel. My name is Dr. Anwar Yusuf Dunbar, and this is Big Discussions 76, Science and Technology. Today is February 27th, 2021. It's the second to last date of Black History Month. And I just wanted to come on and have an open science hangout. Here in the YouTube streets, there are hangouts of all kinds. And I know on Saturday nights, Art of Idmore, for example, they just get on and they just go and go and go. And I have to go to bed at a certain hour, so I'm not always able to hang out with them. But give me a second here. I'm actually streaming this one over to Facebook as well. And I just want to take a look and see what's being seen over there. I'm not, for those watching on the Facebook side, I'm not camming up today. I'm going to go with the avatar. A lot of folks in this sector just go with the avatar, and I'm just going to go with the avatar today. So I see that's going over there. I got a lot of likes before the video, and I think that's because Nikole's coming, but that's also because I think BGS shared it, and BGS shared this broadcast link about 2 days ago, and I want to thank BGS for all of his support in general, but also on the science side. I suspect he'll show up today, or he'll be listening at some point. So there we are. So this is an open science hangout. I have some discussion points at the beginning that I want to get to. Nikole's got the link, so I think she'll come through at some point, and I don't know what she wants to say or what she has to share, but I think it promises to be interesting no matter what. So I'm going to go ahead and share some things and recap everyone on the content I've uploaded on this channel over the last 2 to 3 weeks since the last time I went live. Now, again, I am streaming over to Facebook as well. This is the first time I've done this on this channel. I don't know who's watching today. I was pretty mixed about streaming over to Facebook as well. Okay, there's Nikole in the chat. I was pretty mixed about streaming over to Facebook as well because some of my coworkers are Facebook friends, and Facebook is a bit of a double-edged sword. And so you got to be careful when you're Facebook friends with coworkers. You got to be careful because you just don't know. And so I'm not going to say anything that's related to what I do in my day job. But I thought that for some of the things that we're going to discuss here, it's probably cool for people. It's cool for people in the general public to know, because I'm going to talk a little bit about the vaccine and what's new with the vaccine. So Nikole is here. You know what, Nikole, I'm going to bring you up, and we'll just, I'll talk through some of these things, and if you have something to say regarding what I share, then we'll just keep this informal, and I'll just bring you up. What's going on, Linus?
How are you doing? Good afternoon or good morning? Well, afternoon for you. How are you doing?
Yeah, it's 2:07 here on the East Coast.
Mm-hmm.
How are things?
Everything's good. It's good.
Okay. You know what? I'm always torn between calling you the Lioness or the Scientist Supreme, or just Nikole, or
Nikole.
You've got so many names.
I know. I don't know. I don't make these names. I'm just Nikole. So it's fine. Whatever people choose to call me, that's fine.
All right. I see AB Media there in the chat. What's going on, AB? I see Doug as well. So I'm just gonna walk through some of the things that I've covered here. I think some of these things you probably seen yourself, though I heard you were pretty busy with grants and things. So I'm just going to take a quick walk through some of the videos that I uploaded. I'm going to start with, I'll start with the J&J vaccine. So I was going to give a little talk about inhalation toxicology today, but then last night I saw that the Food and Drug Administration actually approved the Johnson & Johnson vaccine. So I figured I'd read through. I found a nice New York Times article with nice, pretty pictures for everybody that I thought I would read through. But I uploaded this video probably about 2 to 3 weeks ago, saying that this thing is close to being approved. It's one shot and it's 85% effective against preventing COVID. And then I think it's 65% effective against helping people who are already afflicted with the virus. So that's, if you're interested in that, that's in my library. I also did this one. Okay, Nikole, you might laugh at this, but this was in social media a couple of weeks ago. A Gorilla Glue. Actually, that's not the thumbnail for it. Let me find the thumbnail for it. Did you hear about the Gorilla Glue?
Yeah, I heard about it. It was unfortunate that happened. I guess for it to spin out of control the way it did, she really obviously just wanted attention, but it's a price to pay for that. And she hopefully learned a lesson from it. I don't know, but that was wild.
Well, I hope everybody learned a lesson.
Yeah.
Read what's in it before you use it.
Yeah, it's kind of obvious. Those types of chemicals, I would never think to. That's like someone getting silly putty and putting it in their hair as a shampoo. It's kind of obvious. I guess certain things are not as obvious, or reading is not as fundamental as I thought it should be, and comprehension of what you're reading. But yeah, I'm glad that people went in on her actually, because hopefully it would teach other people who are seeking that type of attention. Like I think other people did actually try to seek attention after her, but I don't know. It's just, it really is a testament to the public in general. Like
Okay.
what we do for attention. That's what's crazy to me. But
how bad did it get in terms of folks going after her?
People went. I think I don't think a lot of women went after her as much, but I think people going after her, like just saying like, you're so stupid. I remember I was listening to Art of It War's stream the other day. They were going in on her and stuff, and yeah, that I appreciated that actually. Women, there's no way you can defend this. You defend this, you're defending stupidity, and then you look stupid. So it's best for a female that deals with, as myself, that deals with hair products and stuff. I'm like, what is this?
Okay. Mm-hmm. Well, as a pharmacologist, toxicologist, one of the things, it just showed up on my Twitter feed, or at least I noticed it one Sunday night. I think that might have been Super Bowl Sunday. And then I started seeing people talk about it as the week went on. And I was like, okay, what's going on here with this Gorilla Glue? And so I saw the story. It looks like Tessica Brown has successfully, she successfully, a plastic surgeon successfully helped her get past that. But as a pharmacologist, toxicologist, I said to myself, okay, well, what's actually in the Gorilla Glue? So I recorded this video. BGS shared this one, and I think people were pretty interested in it. So it got a good number of views. And the active ingredient in the Gorilla Glue is methylene diphenyl isothiocyanate.
Wow.
And that's the structure right there for all the curious people. And I tied in that pre-record. This is what I showed before, Chris Rock's good hair. And I never knew that. The other thing I learned about this scientifically was that I never knew that the active ingredient in relaxers was actually sodium hydroxide.
Yeah.
So
yeah. Extremely alkaline, pH is like what, 12 or something.
Uh-huh.
Sodium hydroxide, and we use that in the lab. Very familiar with sodium hydroxide. This is the pellet crystal form, but we have this powder form. It looks like salt, looks like table salt, and sodium hydroxide is no joke. Like, that finding out that was the major active ingredient in relaxers and all types of other products, other conditioners, neutralizing shampoos, and some of the because you can't just shock the hair with different types of chemicals. You have to bring the pH back down. Once you
Oh,
have it up,
wow.
you have to bring it back down again. And especially for people who have kinky, coily, curly hair. So when you're stretching it with this sodium hydroxide, this base, this alkaline chemical process, you're stretching out hydrogen bonds, you're stretching disulfide bonds, well, you're destroying disulfide bonds. And you're making the hair less acidic. You're making the hair less natural, right? That's what you want. You want it to be stretched out and straight. But that is a very harsh, very harsh process. I learned that from the movie Good Hair as well. And I
Okay.
think it was an experiment they did with the I think it was with the soda can in that movie. And it
Yes.
ate the soda can up, completely degraded it.
Yes.
Yeah. Mm.
And if I recall from my lab days, and you may be able to confirm this, but if you dissolve this immediately, it's an exothermic reaction, so the water heats up
Yeah.
pretty
Yeah. Very
rapid.
fast.
Yeah.
Yeah. And that's why they have so many stabilizers in the relaxers. I used to get relaxers when I was younger. And so that's how I know there are a lot of stabilizers in the relaxer chemical. That's why there's a difference between, if you ever go in the pharmacy or grocery store in that hair section and you see the different relaxers, they'll have regular and then they have super. So the regular super is based on how much sodium hydroxide is there. Like super may be like double or triple the amount per dose, per person. And the stabilizers they put there are to keep the exothermic reaction from happening too quickly. You see,
Okay.
because if that happens too fast, I mean, how these chemicals, how these molecules react with one another, it's like instantaneous. It's just so fast. But I've actually had done that by accident. I put too much sodium hydroxide. I was making a 1 molar sodium hydroxide solution,
Mm-hmm.
washing certain type of equipment that we have to wash with sodium hydroxide solution. And I had the pellets, or the salt crystalline, just like this, like in a taller beaker.
Mm-hmm.
And I poured the water in, and it was just like I was holding it with my hand, a gloved hand, and heated up so fast. I was like, oh my God, I had to step back. So I have to do that under a hood in a larger container, a wider, container, with the container on the scale in the hood, because it's not just the heat on the glass, it's the gases released
Mm. Yeah.
as well that you don't see. I didn't even see it. This is before we wear the masks. And I could smell it, and it just burned my felt like it was burning my nose, inside of my nose.
Sounds like hydrochloric acid in a way.
Yeah, and that's the opposite. That's what's so strange to me. See, this happened with the addition of H2O,
Mm-hmm.
but hydrochloric acid is like 2 or 3 pH, and
Yep.
that your nose is burning with nothing added to it,
Yeah.
right?
As soon as you take that top off the hydrochloric acid, you can see the cloud coming out.
Yes. Oh my God. That kills me every time because I have it under the hood and that cloud comes out. I'm like, oh my God, I have to hurry up. And the pipettes we use for aliquoting that out, taking some of that out of the large container, like a liter, and putting it into a smaller container, they have to be glass pipettes because it melts the plastic. You know, if it stays in plastic too long, it starts to degrade like the plastic down. So, but yeah, that messing with chemicals like that. I mean, the women that are getting the relaxers might as well be giving themselves hydrochloric acid because the sodium hydroxide is so pure. You know, I'm sure what they're having in these kits, because that's what it is. It's a when you're doing a relaxer, you're doing a scientific reagent chemical test kit. That's what you're doing on your hair.
Mm-hmm.
You're doing an experiment. Every time you do it, you're doing an experiment.
Yeah.
And so that was one reason why I stopped. My mom was like, there you go again with your experiments. And I'm like, I was being lazy. I was being lazy and I was like, you know what? I got to stop this. Like it took one time for me to get burned and I said, forget it. I had the scabs in my scalp. I said, forget this because I forgot I was not supposed to cause abrasions, which is natural scratching of the scalp.
Wow.
That would be considered an abrasion, just scratching your head. And if there's women in the chat, they know what I'm talking about. You're not supposed to scratch your head for like 48 to 96 hours or so, depending on your scalp or your hair texture before you get a relaxer. And
Wow.
I'm scratching and like that's why you see women pat their head because they don't want to scratch it. Because once you scratch it, if you put a protective barrier on your scalp, such as petroleum jelly, something like that, but it doesn't matter. That doesn't create a complete barrier. Some of that relaxer is going to get through that barrier, and when it does, you basically it's like lighting a match on your head,
Wow.
and it is intense, and so you're sitting there and you can't just wash it all out because then you'll have patches that are straight, patches that are not straight. So you're just sitting there like fanning it. And then you're like, okay, I can't take it anymore. And you go and neutralize it with the shampoo and wash it out. You can't just wash it out. You have to rinse some of it out, I guess, if you have a lot, but you neutralize what's there completely, neutralize it. Then you wash out the remaining amount and you keep washing and washing and washing until the water's clear and your hair is clear.
Okay.
Otherwise, you'll look like the episode where Martin, where Gina was in Shanae's shop and did a relaxer and the woman's hair fell out. She was bald.
Wow.
Like I mean, I'm so glad I don't use this anymore. That's all I have to say. But for her to use that chemical, first of all, the chemical you showed, which is the main ingredient in Gorilla Glue, is another chemical that has that symmetrical chemical structure, which is like very red flag to me.
Mm-hmm.
A lot of the chemicals that are in these different products that have that symmetrical structure, where they're not, I guess, chemically bent, where they have this, what is the word? Not chirality, but they have a lot of oxygen, a lot of carbon at the ends.
Oh,
Yeah,
those
that
that
is. Hmm?
that's not a let's see, that's not a ketone because
Mm-mm.
it's 1, 2, 3, 4, a carbonyl.
You have this is the ring structure. This is what is it? Not
A phenol.
pyrene. Yeah, phenyl ring. Yes. So you have two phenyl groups that are joined together, and those are very strong bonds between them, and then a strong bonds um, at the ends where you have two carboxyl groups at the end. I think the C, yeah, nitrogen and then the carboxyl there. Like that, when I saw it, it looks like a pair of glasses. I'm like, oh no, this is this is symmetrical. That's usually where I'm my red flag. My antennas go up
Yeah.
when I see things that are symmetrical like that. Very strong structure.
And this is used in a lot of things. It's used, I mean, for glue, for adhesives, but also it's used to make polymers and just all kinds of things. And it's just fascinating that she would spray this in her hair, that anyone would. But as a side note, in the early '90s, no, that was the late '80s when we did that. When I did have hair, I did do the S curl as well.
All
So
right.
the whole Guy, Teddy Riley, Keith Sweat, maybe I'll show you guys a picture one day of what I looked like in middle school with curls on top of my head. So.
Yeah, Professor Black Ops said he had the S curl in the 90s. He's bald now. Yeah, I mean, that's the thing. Like the S curl is, I think it's a lower concentration of the sodium hydroxide, but it's still sodium hydroxide, meaning that you're adding more stabilizers to offset the power of that chemical, of that compound, because that's what the sodium hydroxide is, just a compound, sodium, oxygen, hydrogen. But the just like with this chemical, the Gorilla Glue, the strength of those three elements together to form the sodium hydroxide, that's the issue. That's the main issue. Like that's the issue chemically. But when you're looking at it aesthetically, as far as beauty, it's like, oh wow, this is awesome. Yeah, but look how much the hair had to go through to get to that point. Like,
yeah,
destroyed everything. You know what I'm saying?
Yeah. Okay.
Yeah.
So that video is in my library. Let's see, for Black History Month. I conducted this interview a couple of years ago with a gentleman named Dr. Cedric Bright. And if you all know anyone who's interested in going to medical school, if you're in the pipeline already, this is a good read. And I also, as a YouTube content creator, I was thinking, okay, I can also use old stuff, old recordings. And so I cut out a piece of our discussion about how he figured out how to, the volume. There's a lot of volume in terms of information in medical school, and a lot of medical students struggle with that. So this little video is a snippet of how Dr. Cedric Bright, who's the dean of admissions, I think, at East Carolina University, it talks about how he figured out how to assimilate that volume of information. But it's applicable not just for medical school, it's applicable for any secondary education, or should I say post-baccalaureate education you get into. Because once you go past the bachelor's level, at that next level, there's going to be a lot of information to assimilate. So that was uploaded recently. And then finally, where's the other one? Oh, I just uploaded this one for Black History Month. Now, this one is important to me because, when me and Nikole get together and talk, we're kind of in the biomedical sphere, but I like to remind people, especially from our community, that there's more than just the biomedical sphere. And so I uploaded this, I think it was yesterday or the day before that, about the Black astronauts. There are several African-American astronauts who have served in NASA. We typically think about Mae Jemison and Ronald E. McNair. I know Ronald E. McNair because I was in the McNair program, but there were 10 others, 12 others. So I uploaded this one for Black History Month. And I think that, if this is good to know in case you in the listening audience know of a young person who has an aptitude for mathematics or physics or those kinds of things early on. And most of these astronauts, they were either physicians or they were military servicemen or women typically in the Air Force or the Navy, or they actually had training and education in physics. So those are all videos that I uploaded over the last couple of weeks.
Much appreciated. Thank you.
Yeah.
Yeah, they majored in one of my worst subjects.
Physics?
Yes. But congratulations to them because they actually grasped it obviously very well. I was not able to grasp physics because I was like, I don't get it. I don't get the point of this. But I see where it is lucrative if you are a person who understands physics, especially for me. Well, I'll take that back. I would say biophysics. I can grasp that, but there's biology attached to it. biophysics. I get that because we're doing that with different surgical procedures that we're developing, and we have to use biophysics. We have to use a lot of equipment that deals with physics, and I'm learning like how those physical, how the a lot of the rules of physics, how that goes into developing the equipment right
Okay.
now. So
Yep.
I get it, but if it was just physics, like outer space, I'm like, all the math and computation and quantitation that goes into that, I'm lost. I'm like, what?
Okay.
Yeah, that's
What
what I'd say.
what was the most challenging part of physics for you? Because I didn't understand it initially myself.
I would say because I had it in high school
Yep.
and I had it in college. Most of it was over my head, but I think because it was so abstract, I can't pick a specific point. It's been a while ago, but it was just so abstract to me. I mean, people may think that stuff that I talk about, or even for you with toxicology, pharmacology, they may think that's very abstract. But for me, I just was not able to grasp that form of abstract, knowledge. And I know people will say, "Oh, well, physics is all around you, and this and that." I'm like, yeah, you know. But then I'll think like, what about chemistry? Then I slide into other things because it just was not my thing. Like I don't know, and I think if I had to pinpoint it, I would think that one cause is well, two. One thing is the way it was taught to me, and the second thing was I did not put enough effort into studying, what I was taught, and it's kind of difficult if you're not being taught maybe the best way. Then you
Hmm.
kind of know what to study, and I was just studying for the test, really. So,
Okay.
yeah, see, I had calculus, and I mean, cal was fine. I'm like, okay, I didn't even see the connection between like I'm taking calculus because I need it for, I was going to go to medical school at the time, and I was like, oh, I need calculus, I need this and that, and I was like, physics, yeah, whatever. Like, I didn't even make that connection, but I see what you're saying, BGS.
I think the turning point for me was realizing that there was a formula for everything. At
Mhm,
first, yeah, everything was quantitative and
mhm.
everything had a calculation. And I struggled in high school for the first half a year. And then one day it just kind of clicked. Okay, there's a formula for uh, velocity, there's a formula for momentum, there's a formula for all these things. And it's just algebra at that point. That's the
Yeah.
lower level. Don't ask me to do calc based physics or
Mhm.
the upper tier stuff. But I would say that was the turning point for me.
That was a turning point where you got it. You actually started to appreciate it more, and like, okay, this makes
Yes.
sense to me. That's good. Yeah.
But also once I got into the lab at the University of Michigan, when I look back at it, the majority of our devices and our analytical tools, their physics was all in that. Like our 96 well plate reader, that was
Yeah.
I believe that
Right.
was a diode array,
Right. Right.
and using principles like Beer's law, all that
Mhm.
involved, reading absorbances and transmittances and all
Mhm.
those things and exciting electrons. All that's physics. The mass spectrometer, all that's
Yeah,
physics.
so I was thinking of mass spectrometer and the like, well, yeah, you're right. I mean, the real time machine that we have, well, now they have a digital real time machine where it only takes, like, for us, the minimum for a sample is 10 microliters in a 96 well plate. That's
Hmm.
specifically made for that machine to run your real time. And this is the same type of machine they're using for COVID analysis. It's the Quant Quant Studio 3, Quant Studio 5, or something like that. That's what they're using. And they use multiplex machines and all this other stuff, but that's the basic qPCR, quantitative PCR machine they're using. Well, when you're doing quantitative PCR, you're essentially using quantitative physics. Like, you're using all these different things. You're using I mean, obviously physics is quantitative, but you're using like physics, a lot of physical physics calculations. A lot of physics formulas are in there. You can use a minimum of 10 microliters to run a sample. But if you do digital PCR, which is the newest kind that people are using now this year, the minimum you can use is 1 microliter. So in order to go from 10 microliter, it used to be 100, and then it came down to 50, 20, 10. We use 10 to save on reagents. That's the only reason why we do 10 is just to save on reagents. But now you have 1 microliter, and that 1 microliter of sample with 9 microliters of reagents is taken up by the needle of the machine and aliquoted into 4 or 5 different wells, whatever you set up. So you essentially just adding a very small amount of material. The machine sees the material. If you don't add it and tell the machine it's there, it will say, no, you're lying. It will say, no, I don't see it. Because the laser is underneath the plate and on top of the plate. And the eye or the laser is actually in the probe that comes down to take up the sample. And it will look and it'll say, no, you don't have a sample there. Because the height of your volume, there's a meniscus. It has to fit that meniscus. It has to fit that height, the volume sample you put in there. And it just measures it and it says, no, I don't see it. So you add it and then you say, yeah, it's there. Go ahead and do it. Do what you do. And this machine's like, okay, yes, I see that. And it goes and it does whatever you tell it to do. All of that, like now that you're saying what you're saying, yeah, you're right. It is physics. I have to agree.
I worked in a P450 lab, so we were always reading the absorbance of heme. For P450s, that gives a unique absorbance spectra at 450 nanometers. So we were always using analytical techniques for something. So I want to shout out Dr. Vernon Morris there in the chat. He's a atmospheric chemist. If you're going to hang on for a little bit, Vernon, I'd like you to come up and say a few words. But he and I work with him in the DMV. He was very, very big on STEM advocacy, going into high schools and in the DMV, particularly in the district, and doing hands-on science experiments with the kids there. That was very huge. So thank you for being there and checking this out for at least a little bit. I'm going to open it up eventually. I see Art Newstall is here. Peace, Art Newstall. Thanks for coming through. BGS says, without calculus, physics doesn't make sense. Let's see. Lions, time, baby, LOL. Peace. Let's see. Isaac Newton created modern physics and calculus. Be just immoral. Oh, I thought he created Fig Newtons. Okay, we got some humor going on. That was his mama. I'm going to read a few more. You have to use physics to anticipate electron bonds in chemistry under different conditions. Herbert Lewis says, the educational hour. Dr. Morris says 100 plus on how teachers can influence your love and understanding of science.
That's true.
And BGS says, where's my dictionary? This lexicon is over my head. Yeah, right, BGS. This is all cosplay for you. Sorry?
And I was going to ask you a question. So you say you did analysis on P450 with the cytochrome C. With you worked with some things with metabolism from these samples, or you were just looking at the like what was the context of the heme samples you were looking at?
You know what, Nikole? Let's take a quick look before, because I know everyone in the audience wants to hear about the vaccine. That's what I was going to eventually show a quick article from the New York Times on how the new vaccine, the J&J vaccine, works. But we'll take a quick detour. I can just find this. Basic. Because I actually, on my blogging platform, I actually did write a couple of posts on basic research, and I did hyperlink. Yeah, my two first author papers.
Wow. Okay.
So we'll go to this one, which is all public knowledge. Let's see, where's the? Yeah, I'll just stop it here at this one, and I'll do the screen share, which you taught me how to do. Screen share.
I mean, it feels good to be able to share what you're seeing, doesn't it?
It does. All right, so that right there, that's my first author paper. That isn't okay. This is a mouthful, everyone. Ubiquitination and degradation of neuronal nitric oxide synthase in vitro. Dimer destabilization protects the enzyme from proteolysis. Okay.
It's very interesting that you did NOS. I think that's
So okay, I'm going to try to explain this simply. So nitric oxide synthase, there are three isoforms. There's the inducible, which is involved in immune response, which is coincidental since we're talking about a vaccine and those kinds of things. But there's the inducible form, there's the endothelial form, and then there's the neuronal form. And then the neuronal form is involved in neurotransmission and penile erection. And my graduate advisor had a background in P450s. Those are heme-containing enzymes, and the NOSes are P450-like. They exist as a homodimer, which means that it has two equivalent parts. And what my graduate advisor found was that when he treated rats with, there's a drug called guanabens, also known as Wytensin. It's an antihypertensive. He was, and I'll probably unpack this in later content, but he saw that the guanabens drug killed the activity of the enzyme, but also made the protein go away when they did Western blots. But other inactivators like N-aminoarginine, they killed the activity, but the protein stayed there. So there was something about the inactivation by the guanabens, I'll call it drug A, that killed the activity and made the protein basically go away in the cell. And then other drugs killed the activity, but the protein stayed there. So then the question is, what is it about this drug that makes the protein itself go away? And my research was to kind of unpack that a little bit further. And I found that it was being inactivated and basically targeted towards the 26S proteasome, which is a protease. In our cells, there are numerous proteases to proteins and break them down. And it turned out that the drug was oxidizing a key cofactor, which was causing the protein to lose its dimeric structure. So that's what I worked on. So, and all this,
making it less active, making it unable to do its job.
Sorry.
I was saying I was asking if it was making it unable to do its job because it's degraded.
Yes.
No longer. I'm understanding the words you're having here. I know ubiquitination or tination is adding ubiquitin to something. Where you're basically breaking down NOS in cells, and this they have to be in dimerization. When you break that, then the stabilization is decreased.
Yeah. So the trigger, the quote unquote the trigger, because he had to keep explaining this to me over and over and over again, just like the question, because as you know, when you do science, it's about the question. Every experiment is a question. And it took me a little while to kind of connect the dots and say, okay, what's the trigger? What's the trigger? What's the trigger? And it turned out to be dimer destabilization. That was the thing that was causing it to degrade.
Wow.
So
that's very interesting. Thank you for sharing that with us. That's good.
And so it turned out to be, when I look back at it, that was more of it was more of a drug discovery type of project in my mind. It was kind of metabolism, but it was more drug discovery, looking for targets, figuring out how things work in the cell, and what drugs can do down to the molecular level. So everything that happens in terms of the whole animal, when the whole animal gets sick, when an organ gets sick, pretty much everything's happening down at the molecular level. And then it shows on the whole animal level. So yeah, so I plan to unpack all this, in subsequent content later on. But that'll take some planning. So
yeah, I feel the same about some of my work, because I wouldn't say it's controversial. It's just trying to figure out like what to break down and what, I think it would for me, for some of my work, it would probably be better if it's because I have work that's public as well. I'm so like paranoid about putting it out. I think I've sent a few of my works to different people, so they have it. But yeah, just I'm thinking it'd probably be more like sharing the figures themselves, because then it's that's the meat and potatoes of the article for me.
Yes.
I go by, when I'm reading other people's things, I want to see like what do they do, because I can write a paragraph about figure 1A. I can just write, if I have to reference you, for instance, we reference different scientists. And so I go right to their, their work. And from words, especially the title like this, I can figure out what you did. To me, it's explaining it very well.
Okay.
And if I saw the PDF, I'd be like, I'd search for the word results and go straight to the results and like, okay, where's the pictures? That's just how I capture it in my mind. But if I were to explain some of my work, it would probably be in a smaller group that is very interested in that type of study. Because otherwise, people, for me, for my stuff, they probably wouldn't get it. Not in my current work, but a previous work that I've done.
Yeah. Okay. All right. Well, so that was that. So at some point, I'll probably try to unpack some of that piece by piece. I was trying to download the PDF, but it's not doing it for me. So I can show you guys some of the Western blots. But you want to see what's new on the vaccine front?
Yeah, sure. BGS was asking about. He was asking my thoughts on the Israeli exosome CD24 treatment. And I'm still learning about it, BGS. I've actually very strange, but from our RNA-seq data from last month, like CD24 is one of the markers that we have identified that is involved, which I didn't know was involved in exosomes before we identified it. You just have like thousands of genes, and this is one gene that popped up that's high in a cell that we overexpressed the gene in. So we wanted to see, like, if you overexpress a gene, AR, for instance, you overexpress AR, androgen receptor, or you overexpress a mutant of androgen receptor, what does that do to the RNA, the transcriptome? What does that do to the RNA? What does that do to the potential processing of proteins in the cell? How does it affect drug screening, drug resistance? How does it affect all of that? So we did find CD24 to be up as far as the exosomes. CD24 is already in exosomes. Like, they identified that in 2007, and even before that, they just didn't know they were exosomes before 2007. But they did identify in urine, amniotic fluid, and a few other places within the exosomes. So the fact they have this exosome treatment is not as surprising to me. I've heard about exosomes. I've heard about CD24. There are many CDs out there, CD molecules out there. We work with CD24, CD105, CD102. Like there's several hundreds probably. So that wasn't surprising to me. What was surprising is that they have a trial on it, and they're not talking about it as much. That's what was kind of like, okay. But yeah, if y'all want to talk about that or whatever, I just wanted to address what BGS was saying in there.
Well, since we're here, just briefly, an exosome. What's of all the organelles in my library, I don't have a picture doesn't pop into my mind. And if you want to show something at some point, I can give you.
Yeah, I can share my screen real quick.
Okay, so I click. Do I share it with you or can you show your screen yourself?
Let me see. I got my window first. Let me share screen. So you should see when I pop up another screen.
Mm-hmm.
Hold on one moment. I haven't been on here in a minute. Let me see. It should let me do it. Okay. Okay, there it is. So you should see it says CD24 is a marker of exosomes. And
Okay.
when you that screen, it'll pop up.
There we go.
Okay. So they say it in the first sentence. This was 2007. So this was the one I just kept after I started hearing about exosomes and CD24.
Mm-hmm.
So it says CD24 is a marker of exosomes secreted into urine and amniotic fluid. The first sentence says exosomes are small membrane vesicles that are secreted from a variety of cell types into various body fluids, including the blood and urine. So for us, in vitro, you're going to look for the exosomes in the media outside the cells, right?
Okay.
In vivo, you're going to look for it in the blood. So when you have a mouse where you're injecting them with something and they're going to produce exosomes or oncosomes in our case, because we're looking at oncosomes, which are exosomes from, cancer cells. And these vesicles are going to be in the bloodstream. So we have to collect blood periodically from the mice or the patients even. The vesicles, they think they play a role in cell-to-cell interactions. So they're basically little taxis full of proteins, trash messenger RNA, or trash protein that is not that was coded for ubiquitination, that was supposed to be degraded, and it's getting shuttled out of the cell, and it's in the extracellular milieu, in the blood or in the media of the cell that's plated, and we just capture them. You can literally see them. They're pretty big when you're looking at them under the microscope in a plate of cells. You can see them. Oncosomes are much bigger than exosomes, but this is one thing that they went into detail about. I think for this paper, they were talking about glycosylation. They were talking about how they originated these CD24 positive exosomes here. And this is some of their data. They can identify them in urinary vesicles. So this is from urine.
Mm-hmm.
They were able to identify vesicles from that. Because I think that's more compelling because you can quantitate how much is there in the urine from different samples. This is Western blot here. Well, this is old school, but you get the picture. You have heat shock protein 70, aquaporin, you have, which is a vesicle specific protein. We have CD24 protein. And so this is the process that they went through. Then you even have from the samples that they have the negative control, and then you have this anti CD24 samples that are stained with CD24 antibody and positive. Anything that's to the right of 10 to the 0 is positive. Well, yeah, for them, this is positive. It's still low expression, but it's Meaning on the x-axis shifting to the right, it's still low because we usually do 10 to the third and above. But theirs
Okay.
is 10 to the about 10 to the 1.5 to 10 to the 2, which is okay for them, I guess. And it makes sense actually that I think about it just now because these are exosomes, not cells. You see, so exosomes are a fraction of the size of a cell.
Mm-hmm.
So that's why I'm thinking like, yeah, it's probably okay for them to have this low yield of expression. The peak for them is more important. It's almost at 100. It's not like their peak is down here at 30 somewhere.
Are
But
they used now? Is that
hmm?
is that generated using some type of flow cytometry, or how are they generating those peaks?
Yeah, so this is 2007. They probably used an Curry C6 flow cytometry machine or like BD or something like that.
Okay.
Because that's the oldest machine that you can you can use where you get these kind of peaks and this kind of image that's clean. I mean, I'm sure there's more noise than this. They just smoothed it out.
Mm-hmm.
And they have the settings where you can just smooth out your graph, make it look nice and clean like this. But in the method section, they also have, I'm sure, what they did, like what kind of equipment they used, whether it's the LSR2 or, well, Sony wasn't out then, but LSR2, a Curie cytometer, so many different ones, and then they have here images of the exosomes. So they actually do just look like bubbles in the media of the cells. It's not anything special there's not anything special until it's more of them than cells, essentially. But I'll stop there.
Okay. Now, okay. Well, that last statement you made, there's not anything special until there's more of them than cells. Does that mean that if you see more of them than cells, the cells have died, or the cells have undergone apoptosis or something? Or
No, not necessarily. It could just mean that the cells are stressed. It could mean that the cells have um, some type of metabolic issue. They could be resisting a drug because we've noticed that with some of our colleagues, when they do exosome and oncosome work, when there are more exosomes or oncosomes there, is sometimes after they've given them a drug, a very high concentration of a drug. They already know from doing sequencing before, like ChIP-seq, RNA-seq, DNA-seq, genomic platforms, all this stuff. They know that these genes are in this cell is very high, and they know that those genes are also related to drug resistance. You can just do a quick, search online, the different databases to find out your special gene is involved in drug resistance. So if it's high, then the tissue is resistant to drugs. So if you have a lot of these oncosomes and exosomes being produced, it could be the cell is producing this trash or this extra protein for protection as a defense mechanism. It may not be that they have all these exosomes in the media or in the bloodstream of a mouse and it's causing them to go through apoptosis. That may not be it, because a lot of times when they do the apoptosis, I mean, the last figure right there on, I took it off the screen, but they did a Western blot for a Nexin. A Nexin actually went down when they had more exosomes. So a
okay,
Nexin is a death marker. A Nexin 1, a Nexin 5, a Nexin 8. These are death markers. When you have high Nexin, you have high cell death. But when they had more exosomes in the samples in that paper, they had less annexin expression. So it didn't have anything in that case with those kidney cells. It had nothing to do with death as far as them having the exosomes there.
okay. I want to shout out some folks in the chat. We got Nameless is looks like he's listening in. Dr. Tiasan Johnson, welcome. Rael. So we have some of the heavyweights out there. So okay, so Nikole, so we'll just take a quick look at this article because the interesting thing to me, well, I'll show it. We'll show it. We'll just toss it back and forth, and let's see. I'm not going to read it verbatim. I'm just going to walk through some of the pretty pictures. Oh, speaking of which, before we do this, one of the other videos I shot, someone asked me to write something on the vaccine, which we're going to talk about right now. But this person is interested in why people aren't in a rush to get it and why people are skeptical of it. So this is in my library. I think I uploaded this week. But if you want to email me or leave a comment under this video about why you're not going to take it, or you're going to wait until the ninth hour to take it, I'm curious to people's responses. So this was so word came out yesterday that the FDA approved the Johnson & Johnson vaccine. I shot a video on this. The big difference, well, two big differences in my mind are that the Johnson & Johnson vaccine is one shot. The Pfizer and the Moderna, those are two shots. And several people in my familial circle have already started getting vaccinated. My mother's one of them. My father's another one, and other relatives from the baby boomer generation. So far, no severe adverse effects, though if something happens, it's typically going to be after the second shot. And so, yeah, so we have the pretty picture here of the coronavirus, at least one variant of SARS-CoV-2. You see the capsule here with the gene inside and the spike proteins. I'm sure all of you know what those are at this point. Nikole, if you have anything to say, please stop me. So a piece of the coronavirus. So this is, so what I read from this article, or what I understood from the article, is that the Johnson & Johnson vaccine was going to use an adenovirus with the viral DNA inside instead of the RNA.
Mm-hmm.
So, so you have a difference in the, I don't want to say the antigen, because the antigen is actually going to be the spike protein that DNA is going to create. But in a nutshell, Johnson & Johnson is going to, they're using a DNA approach versus an RNA approach.
Yeah, I found that interesting.
Do you, in terms of, what are your thoughts on that?
Because you're dealing with an RNA virus, they're using DNA. They're taking a step back from what Pfizer is attempting to do, what is doing with the RNA-based vaccine. This is a DNA-based vaccine because this is standard. Like many times I've talked about, like, we have a standard of what you do for transfections, because that's what you're going to be doing. Adenovirus, anytime you see the word adenovirus, just know that means transfection.
Okay.
Transferring genetic material from one place into your body or into a body.
Okay. Yeah.
By force. That's what you're doing. So when you have DNA inside of an adenovirus, you have usually it's like a recombinant, or it's a Cre-Lox, or it's something where there's a chemical or antibiotic that has to be in the presence of that adenovirus to get it to be expressed, because that's what you want to do. You don't want to put this DNA inside the adenovirus to get it to just amplify. You want it to express so that it can be detected, right?
Mm-hmm.
Amplification can be something that's short term, where it's amplified for a couple of days and it's gone. It does its job, it has an effect, and it's gone. But you want it to be expressed so that when we have the follow-up from someone who has this vaccine injected into them, we can detect it later on down the line. And I think this is one of the best ways to go if you want to stick to tradition. Now, how they develop the adenovirus, how they configure its structure, what's contained into it, how much is there, as far as concentration of the DNA, that's another story.
Mm-hmm.
But the basic concept of having DNA inside of an adenovirus for this vaccine. That is, for me, as a traditional scientist. I'm not a purist, but I'm more traditional biological scientist. Like, that is very to the book, right to the book. This RNA business, yeah, I'm still on the fence about that. Like, I don't really appreciate that, I'll say.
Okay, interesting. Okay, well, this article, everyone, it's below the this broadcast. The rest of it, it's pretty understandable for the general public. They show, a viral cycle here. They show the virus entering the cell, incorporating its DNA into the host to give rise to a piece of RNA. And the transcription and the translation. Mm-hmm. So basically, the mRNA was transcribed, and now the translation is taking place here at this ribosome to create the spike protein. In terms of immunology, I'm not sure endpoint is the right word, but I'll use endpoint in this instance. The endpoint is the same, to generate the spike protein for your immune system to see it.
Right. But there's no memory of your cells with the other one. There's a memory, like Amos is asking, going about it this way, is it more stable? I think so, because your genes have a memory of, well, not to be too esoteric or out the box. Your genes have memory. They have a ability to categorize, store, edit, like a hard drive. They do what they do, and they also have the ability to, but it has to happen in a certain sequence. They have the ability to regulate what is happening and how it's happening, how often it's happening. Something happens too much, it's like, wait a minute, something wrong. Because the genes your genes are part of nature. Nature always wants to conserve energy. With this way, it makes more sense because your DNA is aware that this is going on. The issue I have with Pfizer, why I said I don't have the same appreciation for, I'm not trying to disrespect them, much respect to them for doing what they did with Pfizer, getting it out that quick. However, my appreciation is lower for that, because with RNA, you're going straight to the RNA, straight to protein. DNA has no idea what's going on. Unless there's a trigger for the RNA vaccine you give somebody to cause reverse transcription, and you make DNA from that RNA sequence, which they did not program it to do. DNA don't know anything that's going on. So, at a genomic level, at a basic genomic level, or at a single-stranded DNA level, there's no memory. It's just like, that's like giving someone a drug. That's why I saw that the mRNA vaccine. The next vaccine will be probably a microRNA vaccine. If you can get people to take an mRNA vaccine, next is microRNA, which is even smaller. The smaller sequence, which is easier to hide when they give it to you. The DNA vaccine, I think, is more stable. I think it's, like I said, more traditional. My mother had a polio vaccine. They still detect it in her Buffy coat.
Oh, wow.
Back when she was a child. That's DNA. That's adenovirus. Once it's there, and if it's made the right way, and which is old school, sorry to say, old school sometimes is better. If it's made the right way, and it has a high enough titer for a certain age group or a certain health demographic in your population, it can last for decades in your system. I'm glad you showed pictures about the transfection and whatnot, and how it gets in and what happens. That's very important.
So, Nikole, when I was reading this last night, and I have to brush up on my immunology. Is this what you were referring to in terms of remembering the virus? The DNA. Were you saying that our bodies can remember the DNA sequence?
Yeah, I'm. Let's see. A single dose of vaccine. Okay. Yeah. Yes. Yeah. It's not just the immune system. It's the cells that are going to be affected, which we don't know. That's random. Lung, heart, liver, brain, kidney, whatever, circulatory system. We don't know which tissue is affected, but those cells in each tissue has memory. At the genetic level, they have a memory. They know, like, okay, this happened, and that's different from immune system memory. Your natural killer cells, your cytotoxic, cytolytic T cells, your different B cells and hematopoietic cells, dendritic cells, and so forth. That's different. They have their own memory as well for that system. But I'm saying your tissues, within the tissues, you have cells, groups of cells. Within those groups of cells, you divide them out. You have, group 1, 2, 3, 4, 5, epithelial cells, stromal cells. You have your different types of epithelial cells that are there. Different types of stromal cells or fibroblast cells that are there. Okay. And they all have genetics, genes within them that tell them what they are supposed to be doing. They have to have memory. They have to know what it is they're doing, which that memory can be adjusted with changes from your environment, changes of things that you choose to do, which Nameless and I have spoken about. That's where the epigenetics comes in. The methylation comes in. The chemical composition changes, meaning that the structure of the DNA changes. So the memory of the DNA changes. That's what I mean. But yeah, like what they're saying here, I agree. Like this is an idea of what I'm talking about. They're specifically, of course, talking about the immune cells. And there is, like, they're saying memory, B cells, memory T cells. Yeah, of course, they retain information, and that's the thing people sometimes miss. They call them memory T cells, cytotoxic, cytolytic T cells, macrophage cells, things like that. These cells have DNA. They make protein. Okay. Your antigen presenting cells, they really have a memory. They have to remember how to present an antigen the right way. If they hold up a sign, the wrong way, the guys that spin the signs in front of the pizza shop, $7.99 large pizza Friday, and they spin the sign. If you don't hold that sign up right and you have it flipped backwards, you just spinning the sign that's blank, nobody's gonna come in there. You have to present the antigen, present the pizza you're selling. There's pizza here. You got to present that sign properly, or nobody's going to come, or they're going to fire you. Well, they're not going to fire you. You're going to be killed. There's a signal that occurs and you will commit suicide. That's what will happen to you because you're wasting energy. You did not present the antigen properly. You got to go. That's what will happen to you. So the antigen presenting cells, they have a memory. They have, and the memory, what I'm saying, is coming from the DNA being regulated by histone modifications, being regulated by methylation in the form of hypermethylation, hypomethylation. The protein that comes from that hypermethylated DNA, which is DNA that's just tied down, really tight and locked down. You can't get protein from that because the DNA is locked. So people say, well, there's protein that can come from that. No, that can't happen. Why did the DNA lock itself down like that and lock the door and have 5011 locks on the door? Because there's some stress, some type of stimulus is causing that DNA to lock itself down. And it remembered from before there was some kind of stress and it did not lock itself down and it was damaged. And you got a mole on your back or something, for instance. This is for instance. That mole on your back that's as big as a quarter, well, yeah, there happened because there was some DNA modifications that expressed itself in a protein on your skin. But that's not supposed to happen. And so, like, I think what they're saying as far as this vaccine is more sound to me.
Okay. All right. This is below in the description box, but I'm going to drop it in the chat right now in case anyone wants to take a look at that. There goes. And I'm just going to briefly show one more. I'm not going to read through it, but as a farm tox guy in regulatory affairs, one of my chief concerns is the safety. So I found this article as well. So I will probably do some more investigations on this. This article says J&J's COVID-19 vaccine is safe and effective. FDA says, paving way for approval. So we know that just got approved yesterday. So I'll be creating some more content on the safety. I'll probably pull the memorandum down from the FDA website again and take a look and see what I see, though I suspect it won't be significantly different from the Pfizer and the Moderna. But I'm going to drop this link in the chat as well in case anyone else wants to peruse through it. And that picture is from someone in South Africa. I was looking at the, she's got looks like she's Indian because she has that dot on her forehead. That's in South Africa. So with that, Nikole, do you have anything else you want to share before I open it up, or anything you want to say? Or
just quickly, I just put a link in there for the clinical trial. So this is a clinical trial where they're setting up for the exosome CD24 treatment in patients with COVID-19. I just posted that in chat just now.
Okay.
That's all.
Okay. Now, what's that about?
So it's the open label. This is a phase 1 study.
Yes.
They're still recruiting right now. It was posted February 10th. So they don't have the dates of when it would be done, as they're still recruiting it. But this group has started early, and they are coming from Tel Aviv. So this is Israel.
Oh, okay.
So it makes sense. People want to jump on this pretty quickly. The contact information is at the bottom of the link at the bottom of the screen. When you click on the link, their email is there, everything as usual when you look for a clinical trial. But the important thing for me is the green box. And it doesn't have anything as far as like when it's going to be done. They haven't started yet. So the best thing, if you're going to follow this CD24 exosome treatment, is to look for that title on NCBI, or even search on Google Images for that phrase.
Excuse me. So the exosome. So in terms of a therapeutic strategy, is it designed to export the viral particles out, or?
From what they're saying here, they said that CD24 is a small, heavily glycosylated, so it has a lot of sugars on it, and it's a GPI anchored protein. It is a key player in many cancers. So what they're saying here, because I know it's very strange, I would say read background on CD24 and exosomes separately and then converge them together, because CD24 is high in cancer. But a lot of papers recently being like from 2015 on, have talked about the similarities between cancer cells and viruses.
Okay.
And so if you just keep that in mind, it'll help. But they say that what they want to do is, they literally say the rationale for this treatment is that exosomes overexpressing CD24 that are isolated and purified from Trex 293 cells, they're engineered to express CD24 at high levels. So Trex 293 are basically like the cousin of HEC 293 CBI. And so these Trex cells are good for modeling T cells. And they're specifically engineered to express CD24 at high levels. They basically just CRISPR them and put more CD24 in there. That's all. So they can suppress the cytokine storm. That's the problem that they're having in Israel, at least. The cytokine storm that's killing people because their body is in shock. So they deliver them directly to the target organ. These people already have COVID. Maybe it's in their lungs. You give them the maybe an intranasal or something like that, or an injection directly. and using the exosomes as a highly body compatible delivery vehicle. So you're trying to deliver these cells that have, you're trying to deliver the exosomes that have the CD24 exosome treatment there into that organ specifically. So it only affects that organ.
Mm-hmm.
What they have to do is they're going to have exosomes, the exosome / CD24 vesicles that are specific for each organ. And there's a way you can do that. There's a way you can make it so all you have to do is look up the sequences for CD24 in the heart, CD24 in the lungs, CD24 in the liver, and so forth. And the sequence is slightly different. Maybe this not going to be different as far as differences in the AGTC pattern. It'll be different in how the protein is made. Because CD24 may not be as highly glycosylated in the heart as it is in the lungs, for instance. So you have to make the vesicle fit for the heart. Maybe there's 50 glycosylation patterns on the heart CD24, but the lungs maybe has 150. So you have to this is in the public, this is what we call precision medicine. That's what they're doing.
Okay.
It's precision medicine. It's almost like patient to patient in a way.
Right.
But I expect that from a lab in Israel. Like I kind of expect that from them because they like to do things like one time, knock it out and get it done. Like their endpoints, their outcome measures are like they're very succinct about what they're doing. And that is much appreciated. I mean, the page, when you click on it, is very short.
Mm-hmm.
Most of them are probably 5-6 pages long. This one is maybe 2 pages.
Mm-hmm.
So, and this is the phase 1. So when the third, they have like phase up to phase 3 or something, but this is safety. They just want to see if it's safe. So they're going to go through a lot of trouble to make this vesicle packaging program and then see if it's safe. If it doesn't kill, make sure it doesn't kill anybody.
Okay.
And I think, I want to say one more thing. This is what they should have done with Pfizer. This is what they should have done with Johnson & Johnson. This is what they should have done with Moderna, Cambridge. Well, Cambridge probably did. I'm not sure about Sinovac from China, but this is what many of them should have done. Why are you testing giving a virus to healthy people? You have all these people in the hospital that are sick at different stages of the sickness. Why are you giving it to healthy people and then turn around and give them a treatment? I don't understand that. I mean, maybe I am ignorant on that front about, about how that works, but I was like, I don't get that. Why don't you give it to people that are already sick, just like they're doing in Israel? That's what their plan is to do. Give this treatment to people who are already sick. You already will sign documentation, clearly, waivers and things like that, which is what we do normally in clinical trials. Well, I deal with people who have cancer. They've had it for decades. You best believe they're going to sign a waiver. That blew my mind. I said, healthy volunteers. I'm like, what the hell? I'm sorry. Like, I was like, what? I was like, what this thing is? Like, clearly that tells me. I was like, you don't know what this is. Like, you really don't know. Okay. All right, Cletus. All right. My bad, Dr.
That's okay. That's okay. You're adding to my body of knowledge here. So, okay. Well, everyone, this is an open science hangout. So I'm going to drop the link in the chat. If you haven't subscribed, please subscribe if you want to make a donation. I left my Cash App there, a few chats up, a few messages up. Nikole, once again, I want to thank you for coming and gracing my channel again. It's always an honor to have you here. I want to drop the link in right now. Vernon, if you're still out there and you want to come up and say a few words, please do. But if anyone wants to come up and say a few words and ask questions and whatever. The link is right there.
Mm-hmm.
And so, in as much as you can reveal, how's the gene editing going?
Oh, the
Your research, yeah.
Oh, well, we have a lot of it done, but right now I'm just living off the wave of getting the grants a little bit.
Okay.
The hype around that for myself, Um, yeah, the gene editing, I'm not doing as much with gene editing. I was doing the RNA sequencing recently, analyzing the sequencing data and learning about because I remember mentioning back in like December, I was learning about how to analyze my data
Mm-hmm.
and how to use certain platforms like the gene set enrichment analysis, going to classes, learning how to see my data better, how to present it better. So we were just doing. I mean, this was 2 days before New Year's Eve. I had to submit samples for RNA sequencing,
Wow.
and yeah, and so I submitted all that. Got that in. That was analyzed like maybe end of January. It was done because we were in a queue behind people that have been backed up since the fall of last year. And that's all analyzed. And we know that we have specific gene sets that are higher in a cell that we overexpress. We didn't overexpress this gene using CRISPR. We did this without CRISPR. We did a CMV promoter, the old school way, which is more beneficial to us because then we don't have to like mention CRISPR at all, which is good.
Mm-hmm.
And we did that. We could just mention ourselves, the hard work that we did, and that's good. That's the first paper. The second paper is there was some CRISPR there. I think no, the first paper does have CRISPR actually. It has two other cells that have CRISPR, two cells that don't, two cells that do. The cells with CRISPR that I use personally are all knockouts.
Okay.
Put anything in to make the cells worse than they are. They're already jacked up. I don't need to jack them up anymore, make them worse. So I knock out certain genes. CD105 is a gene I like to knock out. And many of his compatriots that help it to be tumor promoting gene, I knock them out. But right now we're just looking at how the epithelial to mesenchymal interaction, the cells that are epithelial, how they interact with the cells that are mesenchymal. Mesenchymal cells are more stem-like cells.
Mm-hmm.
Epithelial cells are more they're forming the structure of an organ. And so the mesenchymal cells are more likely to influence the epithelial cells to be an aggressive tumor, be more aggressive. They're like bullies. They try to jump the epithelial cells into the gang. Okay, that's how I see it. And so we just said, okay, we'll just take the fibroblast, these mesenchymal fibroblast cells, we'll just knock out the genes that they use to do what they do. And now see what they can do. They still grow the same, but they don't have the same, they're not on the block as much, you see. They at their grandma now because they on probation, they on parole, you know what I'm saying? They done got shot, or whatever. These are the similarities I make for myself. My analogy. You was a gang member for 20 years, you got shot up, you've been in prison, now you at home. Man, I ain't moving the same way no more, man. I paid in full, I got shot. I can't even move the same way, that type of thing. That's how I feel about it. They don't move the same way, literally, doing motility assays. They don't act the same way genetically. The genes are up and some are down. They cannot interact with the epithelial cells. They don't even recognize them. When we put them physically put them together, they are like, I don't know what I'm supposed to be doing. I guess it's like they have amnesia or something. But that doesn't last long because these cells, they always find a way. So when over time, with about maybe 100 generations, then they start to remember what they're supposed to be doing. I'm supposed to be an aggressor. and they go back and remember that, and they start doing what they were doing before. But in the meantime, what we do is we put these cells in mice. We block their ability to metabolize sugar, block their ability to metabolize glutamine amino acid. So that's the second project. The project for the grants is totally different. It's dealing with aging. How can you block aging from happening within certain organs?
Okay.
By manipulating the gene sequences. So that's what I got the money for. Was one was cholesterol. We can target cholesterol. We can target tumor cells by targeting the cholesterol they need to make steroids. Because if you give prostate cells, for instance, which has been shown for over 15 years now, if you give them exogenous cholesterol, they will take that and process it through their enzymatic reactions, through a lot of the CYPs, and CYP17 beta and all these different things, and testosterone, dihydrotestosterone, all the different types of testosterone hormone. They'll process the cholesterol and make their own steroids. Even when you block the steroid in the media, you block it, you give them charcoal strip, media, but then you turn around and treat them with cholesterol, certain amounts of cholesterol. Over time, they'll make their own. They're like, okay, we can make our own androgen. We don't need you. And so they do that very well. And I was like, okay, so what does that mean for their metabolism? That's what we need to figure out. Like, is it causing them to age? Is it causing, if the organ's aging, does that mean the tumor's getting worse? These are the questions we're asking. And the final project is on aging from an epigenetic level and looking at, trying to look at how fat, not cholesterol, but how fatty liver induces aging. If you have a high fat diet, you will induce biologic aging.
Wow.
If you have a lot of fat is number one. Sugar, not so much, but fat is number one. If you have a lot of fat in your diet, you will induce biologic aging, not chronologic aging, because you can't, we don't control time yet, I guess, but we don't control time. But you can cause aging to occur in the cells. And aging in the cells is just Michio Kaku talked about how aging is really the cell getting tired of doing these processes, where it has to constantly make sure the telomerase is there, the enzyme is there, make sure these telomeres are a certain length, make sure that it keeps an eye on how much methylation is occurring. And the cell is just like, I'm tired of all these revolutions. That's what these cycles. I'm tired of these cycles. Cells don't have a long lifespan. And if they do things to promote aging, which is what a lot of cancer cells are doing, if they're promoting the aging process, it's a paradox, because they're supposed to be dying quickly. They're not necessarily dying that fast, but they're able to reproduce themselves quickly. You're reproducing every single time cancer cell reproduces more cancer cells, they're aging faster and faster and faster. And while they're aging faster, you are dying. You're aging as well, biologically, chemically, physically. You're aging, but you don't see it from the outside. You look like a 35-year-old man, but internally you're 68. So that's what my the last project is. That's the most important one for me, because I can leap into I can transition from cancer into prostate cancer into another organ, which is liver. And I can also transition at the same time into tissue regeneration.
Wow.
So I think that's, especially now with COVID, I think I was thinking about it last year, and I was writing the grants, had all that time but on my hands. I was thinking about it, just sitting here looking out the window at home, and I said, I need to do something with tissue generation, because all these people dying from COVID, and people who are not dying, who have the organ failure, permanent damage. So we need to rebuild their tissues. At some point, it's going to be very beneficial. But I can't just go in there because there's already an industry for it. I have to kind of slide, you know, slide your way in there.
So are you referring to something like kidney or liver or?
Liver would be my primary organ. I would do because liver is the only organ that can regenerate itself
Yeah.
without any kind of like direct stimulus. It just does it. So that's like the life organ that we're looking at.
So going back to the fat, are there specific genes that fat is turning on? Is
Yes.
it okay?
Yes. A few of them are involved with cilia production. Some of them are involved with um like the what is it called? The mitosis, just standard mitosis. Some of them are involved with cell cycle. And so if the fat is there and it's increasing the cell cycle process, you have these cells that are being produced, you're having high cell proliferation because your own genes are producing themselves. So then you have a thing that's called, like, for instance, there's chemotherapy addiction that tumor cells have, where if you take them off chemotherapy, they just die, they crash. The same thing is true with certain cells. If you have them on a high fat, high cholesterol diet, then you take them off. I mean, just not even slowly wean them off, but just deplete them completely, they crash. So there are genes that are changed. Some are with the membrane, some are with the mitochondria itself, which is the powerhouse, energy house of the cell.
Mm-hmm.
It just depends on which kind of cell you're looking at.
That's interesting.
Yeah.
And so I never thought of aging. When I think of aging, I think of time, so chronological aging. But so there's a separate aging from that.
Yes.
Interesting.
Yeah. Yeah. I think BGS sent us something about it. He sent us the article, NMN, different nucleotides that they're using for aging. So sometimes I read stuff and I read into it and I'm like, oh, I could do something with aging on this. Like I could do something with, tissue regeneration. Oh, this is interesting. I see like one or two sentences and I'm like, oh, you know what? I'm gonna look this up. And I just like find a breadcrumb, so to speak, and then go from there. But yeah, it's an interesting field. Like no doubt about that, because the biologic aging explains the dormancy of certain cells, the senescence, stem cells. It explains a lot of different things that happen. Transplant rejection, all those types of things are explained with biologic aging, not chronological aging. You're
Okay.
depending on what you're doing, you may have a heart that's, you're 27 years old, your heart is 27 years old. But depending on what you're doing in your lifestyle, your liver is 48 years old.
Wow.
Your intestines are 21 years old. Like um that's how, work. Your stomach is 48. Your stomach and liver are jacked up, messed up. And that can affect you later on in life with a lot of digestive issues, circulatory issues, immune system issues. If your digestive system is not good and it continues to remain that way for a long time through your life, that affects your immune system. You know, so that those types of things I'm looking into, like how the organs are different. How are they talking to each other as well? Because more than likely, when people are living longer, the longer you live, the more likely you're going to have cancer. No matter what you
Yes.
do, no matter where you live, no matter who you are, the longer you live, that's an age disease. So the older you get, you have higher chances for this biologic aging, of course, and you have a higher chance of tumors, for instance, leaving from, like, for us, leaving the prostate, going to the liver. Well, cells don't decide just to leave from one place and go somewhere else. They usually like where they are.
Mm-hmm.
So something is causing, they call it the homing mechanism, where the liver is sending signals to the prostate like, hey, it's cool here. You guys can come here, have a nice little vacation rental, summer rental. Come here, have a good time. It's cool. Our rates are low. You know what I'm saying? Like the prostate cells, like, let's pack it up and go to Beverly. We're going to Beverly Hills for the summer. They leave, pack up their bags. 100 of them, maybe 100,000 of them leave. Only 100 make it because they die along the way. They run out of energy. They didn't pack enough food. They were not in a aggressive state as they should have been to make that journey. From the prostate to the liver is a long journey to make for a cell. Other cells within the bloodstream, like the police, the immune cells, catch them, arrest them, kill them, and they're gone. So you have a very small percentage that make it to that secondary site. When they get there, they're not going to make the journey to go back to the prostate. And they just saw their friends get killed off and everything. They're like, nah, we good right here. And plus, we're old now. You see, from the prostate to the liver, the cells have aged. They have become more experienced. When they get to this secondary site, they actually begin to form micrometastases a lot of times.
Wow.
They begin to build up. They become stem-like, which is where their proliferation skyrocket. And they start producing more and more and more. And the first generation of them that got to that secondary site, they die off quickly. They make their few colonies of generations and they die off. But they teach all the other cells that come after them what they have learned, which is totally different from the prostate cells that are still in the prostate.
Now, Nikole, is this analogous to cells becoming immortal?
Well, yeah, they would be immortal at that point. Yes, because they could leave from the liver and go somewhere else. We have cells that leave from the liver, go to the brain. There are prostate cancer patients that have prostate cancer in their brain, and we know they're prostate cells. They're immortalized in a way, which is why it's hard to target them, because if you target them, you are going to kill the organ that they're in.
Mm-hmm.
So we trying to develop one of the grants is like developing a drug that only targets the cells that are in the liver that have metastasized to liver. Only target those cells. Don't kill the liver, because the person needs the liver to live,
Yes.
And this is regenerative tissue. So you kill something off in the liver, it regenerates that area where that was. And it's a healthy liver again. No problem. It's doing its job. It's being, it's flushing out toxins and doing what it's supposed to do. The issue is that the liver does not see those cancer cells as toxic. It sees them as, oh, well, you guys are just visiting. So, you guys can hang out. That's cool, And they're like, yeah, we gonna hang out. We ain't never leaving. And they just stay there and destroy that lobe of the liver.
Mm-hmm.
That liver until it looks like you have liver cancer, but you don't have liver cancer. You have prostate cancer, for instance. You don't have even lung cancer. Even some lung cells leave and go to liver.
Wow.
That is definitely an immortalized state, because they can do whatever they want. They can mutate however they want. They can leave if they want to. They can move around different parts of the liver, just whatever they want to do. And as they're moving, they're leaving behind genetic information. The hepatocytes, they're not naturally programmed to be as defensive as they should be.
Okay.
They just signal the immune system. The immune system is like, okay, we'll take care of it. It's like, for some reason with the liver, the immune recognition software, it has an issue, it has a problem.
Okay. I want to thank Nikole. Thank you for the Cash App. Thank you, AB Media, MLR, Master Teacher. Thank you for the Cash Apps. Since we'll go for, if no one else comes up, we'll go for about another 20 minutes. Well, since we're here at the liver, I was looking at this picture and I was thinking, based upon the fact that the liver one of its key roles is cleaning out and acting as a filter for our blood. So all the blood is going through the liver, basically. So it's like a main hub. So it makes sense that other cells would be able to easily migrate there. My question is, since we're talking about tissue regeneration, as you know, one of the organs that science has really wanted to be able to figure out how to regenerate is the nervous system, especially when people get paralyzed, like Christopher Reeve.
Right.
So have they been able to identify what it is about the liver cell types that allows it to do that versus other cell types in other tissues? Are there specific genes in there that allow it to regenerate, or are there specific growth factors? Anyway, go ahead.
There are. I can't remember all of them. I have a heat map on them. I can't remember all the genes, but yeah, there are groups of genes that have a specific function as far as helping the liver to regenerate itself. There's stem markers, mesenchymal markers, and these are the same markers that were expressed at high levels before we were even a fetus. We were just an embryo. So during the embryogenic development, during blastocyst, get gastrulation and all that, when you're developing as a small zygote and then into a fetus, into a baby, that embryogenesis, those embryogenic markers are very, very high, obviously. Otherwise, you would not have been born. The problem is, like, later on in life, when there's some type of disease going on, even without a disease, the liver tends to express some of those stem markers. The problem is the level of the markers that are expressed. If they're too high, there's something going on in your body. And usually if they're too high, you may have cancer, you may have cirrhosis of the liver, you may have what is called NAFLD, which is non-alcoholic fatty liver disease, due to high fat diet. So you can age your liver yourself by having a high fat diet, and those genes that should not be on will start turning on. A lot of those genes are extremely dangerous for the liver itself, because they are on, and the liver starts to delegate whether it should process the fat this way, or should I just process everything at once, or it forgets how to be a good toxicology screener. It forgets how to screen out the toxins the right way.
Okay. Interesting. So basically stem, say that again, stem, I don't want to say stem cells.
Yeah, stem cells.
Stem cells, okay.
Or, some people, I'm using two different terms because some people will find things that have mesenchymal cells and they're like, oh, Nikole, why you didn't say mesenchymal cells? Like, why you didn't say mesenchymal? And I'm like, okay, yeah, I know, but there's two different words, it's just synonyms, basically, the stem cells versus mesenchymal cells. And these are just cells that they want to remain in a state where they can reproduce. You don't want cells reproducing consistently, just all constantly reproducing, and you're a grown person.
Right.
You are already formed. All of those windows of development have closed. Why are your stem cell markers are open the windows again? Let's just go through the cabinets and see what else is in here. No, you're an adult. What are you doing? You're reversing back to a child. Fat causes that reversal. High levels of fat. Specifically, of course, a saturated fat. Very high levels of saturated fat, high levels of animal fat, high levels of animal protein that contains dioxin and other chemicals and toxins that your liver finds hard to flush out.
That's fascinating. Do you know if when the fat gets in there, is it activating a specific receptor? Is it Is it modulating something?
Yeah, so there are several receptors. Like one of them I'm looking at from myself. You have Bmp receptors, and I know it's going to sound strange that I'm talking about liver. The Bmp receptor, Bmp stands for a boy, B meaning for boy, M for Michael, P for Peter. So it stands for bone morphogenetic protein. I know that sounds strange. We're talking about the liver, and I'm bringing up a protein that's bone, dealing with the bone. That's what we identified. But BMP markers, BMP receptors are found all over the body. And
Yes.
usually wherever they are expressed at high levels, there's a problem. You know, you're having issues with metabolic processes, cell signaling processes. Cells are not able to signal properly to each other. What I mean by properly is it's not that they're not signaling, it's that they're talking to each other on a consistent basis, and they should not be sending those messages back and forth. And BMP is one that promotes that. You have other signaling pathways, huge signaling pathways, TGF beta, insulin-like growth factor. You have EGF. You have not alk phosphatase, but alk phos. You have the different ALKs. You have SMADs. There's so many different protein markers. The PI3 kinase, which is also linked with AKT, and these are all things that are involved in either promoting stemness when there's a mutation within the pathway, or blocking stemness, which is usually what you should be doing as an adult. The switching on and off of these genes is where you have these problems.
Okay.
Too many switched on at one time, too many switched off at one time, and the wrong one switched on at one time. And we call them gene bullies. Like when I was talking about the cells that can be bullies, sometimes you have gene bullies where you have a gene that's on constitutively active, meaning it's on all the time, and it's supposed to be turned off. And since it's on, it's supposed to regulate somebody. And it's not regulating who it's supposed to regulate. It's going to jump over them and regulate somebody else, but to the point where it shuts all of those processes down. That's a gene bully. You're like, well, I don't want to tell him what to do. Hey, you over there with the glasses, give me a lunch. And you run over there and beat them up. And then everybody else that has glasses and lunch money, they put their glasses in their pocket, like, they shut down. They're shut down for the rest of the day. They're bumping in the lockers and all this because they're afraid. I don't want to get shut down like him. That's a gene bully for when we talk about how these genes interact with each other, how these proteins interact with each other. But that's a mutation. That should not happen. You should not be running around, knocking out all these other genes. Because when you knock down that one gene, you just killed a process for the cell. You killed a process. That cell may not be able to make exosomes and process waste like it should. If we go back to the exosomes discussion, maybe it needs to push exosomes out because it's trying to get rid of junk proteins and junk pieces of material, enzymes that doesn't need. You just made their trash collection process stop.
Right.
I think that AB he said the link was never dropped. I don't know. I wasn't looking.
Let me try it again. What stood out to me about what you said about the BMP being a bone receptor, that's actually in the pharmacology world, in terms of drug discovery and toxicology, that's something that's problematic because the receptor types are expressed all over the body. So it's hard to make a molecule that's specific for one tissue type because those are expressed all over the body in many instances.
Yeah. Well, we know that in at least in cancer, we know when you have the tumor that has metastasized to the liver from another site, that the liver tumor niche, the tumor has to be in a certain spot of the liver. We could detect that. So if we do the experiment with mice, we just put luciferase in the cells, put the cells into the prostate, wait 4 weeks, and check with luciferase to see if the liver starts to glow. You can literally see them there populating in the liver after about 4 weeks. It takes some time to get there. But if you give them testosterone, if you give the mice testosterone like orally or something, or injections, it might take them like 2 weeks to get there because it's just gonna, promote that movement, that growth faster. So they get there and then they're lit up in the liver, and now you can start to determine, okay, what's really going on here? Like, are they going to respond to this drug? We give them a drug, like, well, the drug we're developing is just not going to do anything to the liver, even though the liver is not the same as it was, even though it is more of a, I guess, quote unquote, it's not naive. It's not a naive liver. It's a learned liver now, educated liver. It knows something wrong is here and needs to get out, but it does not have the machinery to kick those cells out because it still sees those cells as self, host. And the liver knows that there are cells that if a cell of you, which you are the host, if a cell is of the host, I can't hurt that cell. My job is not to hurt cells as a liver. My job is to process toxins, chemicals, anything that you're eating and drinking, anything that you're inhaling that gets this far. That's my job. My job is not to kill these cells because I see the cells as the host, as self. So that self identification, like removing that from them so that they can just be like, look, these are your enemy. They're not your friends. You got to get rid of them. If they can't do it, we give a drug that only targets those prostate cancer cells. If those prostate cancer cells are mimicking a cancer patient and they have high expression of certain genes, well, there you go. The drug will work. We know it will work because we do it in vitro. So that's how we would do it. And that's like, the liver will not be touched. It can still go on doing what it's doing. The tumor will shrink. Like, this is my hypothesis, y'all. This is not, I haven't done it in the mice yet. That's why I needed the money. But, but in order to get it done, like, this is the hypothesis that this will work. It will shrink the tumor. The tumor is gone. Now liver can do what it's doing. And it will also prevent any cells at the primary site. They will be dead too. Because this is an intravenous injection. This is not, anything that you're taking orally. What we have developed is not orally available.
Mm-hmm.
Has to be given by injections, and so that's what we're doing. We want to do this grant because we want to develop a trial for safety at some point, another trial to see if this drug will work. But we pick the genes we want to block, and we know those genes are not in the liver. We use NCBI, genome browser, all these different mechanisms, and we can see, oh, the liver doesn't even have that gene. Okay. Let's use that gene to block the tumor cells. That was it. That's the basic premise. I'm making it in a nutshell, but that's the basic premise of what we're doing.
Okay. Everyone, I dropped the StreamYard link in the chat from the backstage, and let
me try it.
I don't know if it's going through. I went over to the YouTube side and dropped it in again, and I can see it there.
Yeah, they can't see it. I can't see it on the on StreamYard or YouTube.
That is strange.
I'm looking like, where is it? I thought I saw it, but I think that you typed something earlier. I saw
yeah, it's said right in there.
Wall Street Journal, my bad. That was Wall Street Journal.
Okay. It's right down there now. Well, I'm seeing it between Rael's comment and Nameless's comment.
What?
But I don't know.
It's not there.
It's not? No. Try it here.
I put it after Nameless's comment.
And it didn't go?
It's there. I see it on the I see it on YouTube. It's there. And I see it. Okay, I see yours on StreamYard, but I don't see yours on YouTube. This is
I
crazy. I don't understand. Wow.
I've seen this happen to BGS sometimes.
I would say
one more time. And I just dropped it on the YouTube side. Don't see it.
yeah,
Okay. I don't know what's going on.
Oh my goodness. They may have to they may have to
put a one in the chat if you can see the link.
Yeah, another person put it.
The angry man thing. He does that. You know, he says, "Put a one in the chat if you want to hear me talk about Bitcoin."
Bitcoin.
Email it to me.
I need 20 more likes. What's going on?
Hold on, BGS.
Okay, shout out to Angry Man. Oh, he said email it to him. Okay.
Yeah, I'm gonna email it to him right now.
Yeah, that's hilarious. He reminds me of my cousin so much. It's just, it's so funny. And I sent him one of his videos. He was like, "That's not me." I'm like, "That is you. You read just like that. You laugh." And he's so funny. He's my cousin. Like every time I see Angry Man, you know, I'm just like, "Oh wow, that's my cousin right there."
Let's see.
Personality wise, you know, looks wise, everything. I'm like, oh wow.
All right, BGS, I just emailed you the link.
Yeah, he'll come in soon.
Yeah, I was wondering. I was like, wow, nobody wants to come up today.
They don't want to talk about this real. This that real stuff right here.
Actually, no, that's the. Hold on, BGS. That was the wrong one. Let me. Okay, StreamYard. Okay. There. See if you can see it now. Okay, so let me hold on, AB. Let me see. Emailing this to everybody. AB's is AMPX. AMPX 2083. 83. All right, AB, I just sent it to you. Ah, there's the master teacher. Hello there.
Hey, folks. I just want to commend you guys, man, because I've been listening to Dr. Sapolsky from Stanford, and you know, because he's a he's a biological neuroscience and scientist, and he explains these complex things. And I can say reminds me of sitting, you know, watching his class, you know, the way you guys break down things. And you can't find, I can tell you, you can't find this kind of analysis anyplace else on YouTube, you know, as far as we're us actually knowing who you guys are. And I was explaining it to somebody else, and they were saying, well, this doctor, that doctor. I said, no. I said, I know these people. I trust these people. And you guys were way ahead of the curve, way before any of this stuff was published in papers and stuff like that. Everything that you guys said was like 2 or 3 months later. Well, yeah, it popped up. Yeah, we already knew. Okay, we already knew this stuff. So I don't think that people in this space know exactly the worth. Just you guys being here and explaining this stuff it's amazing to me. Because you'd have to pay like $20,000 a year to go to Stanford just to hear this kind of stuff. If you can get into the class, it would probably be a postdoc class, either a postgraduate class or a postdoc class. You couldn't hear this stuff, in undergrad. So, I want to commend you guys, and I want people to understand exactly how valuable you two are. That's the first thing. And I want to say congratulations to my girl. I was telling Dr. Johnson that you really are the shit. Huh?
I didn't see that. I was I'm in my mouth, but
because he was saying, how did she get this grants? And she's a one black woman and black men don't get it and stuff like that. I said, you understand, Dr. Johnson? Nikole is a shit now. I said, anybody else maybe? Anybody else maybe, but her? No. Okay.
Yeah, I understand what he's saying, because a lot of the grants that the black male professors try to go after, they don't get them. And I'm like, what? And I know they're overqualified to get these grants and stuff, especially in Dr. Hassan's field. It's difficult because it's like a block on male professors to get anything like that. And what he's doing, I really commend him on what he's doing as far as getting the information out there. My field is different, but you have to remember, like, I would be a small fish in a huge ocean.
Okay.
If I was still doing what I was doing years ago with breast cancer, because it's breast cancer.
Yeah.
With prostate cancer, there's a different angle to it, because you have veterans. Have literally given their lives sometimes to defending this country, and they're just treated like crap. And I'm like, I'm tired of this. Like, what the heck, So I was like, we need to have some type of difference in what different way in which we're looking at these projects and doing them. Luckily, we have Department of Defense. We have all these different vehicles to get the work done that pay pretty well. So I was like, let me do this so I can get it done. I need the money in order to get the patients the help they need. It's not for me to go buy a car or something. It's to get the patients the help they need. And that's what I focus on. So I would say, to Dr. Johnson, that, I know I'm a woman and all that, but I'm doing it. This is not affecting women. Like, this does not benefit women, this project.
Yeah.
At all.
But you are.
They're not patients.
But you're extremely qualified. So I don't think you know how smart you are sometimes.
Yeah, I'm just doing me.
Yeah, I know. So that's it. I don't think you really know how smart you are, and I think Dr. Dunbar would say the same thing.
Yeah, I mean the grant funding process it's fierce.
Yeah.
So you have to be
on that.
The best of the best to get your grants funded.
Yeah.
Yeah. And I'm glad the first time I was like, yes, I ain't gotta put this thing in no more. I ain't gotta write this thing.
Yeah.
Dr. Dunbar knows, like, you can write a grant and submit it, and you submit that bad boy like 3 times and finally get it. I know he's been submitting grants for like 3 years.
Oh wow.
Grant for like 3 years, and we're like giving him suggestions on how to change it, how to make it better, how to innovate. Helping him get a couple of patents under his belt, so he's more in publications and things, so he's like more eligible and more attractive to his funding source. He has not been able to get his money. I was like, dude, like, He said, I'm either gonna have to go to a different funding source or like go private or something, or go for a grant that's less money. He's going for a $2 million grant. I was like, that's not that high. $2 million for 4 years is not that much. But
Not when they're throwing around trillions right now. No.
right, yeah, right. That's a drop, literally like a couple of drops in the bucket.
Yeah.
That evaporates quickly. Like it's nothing. But he was so worried. He was like, "Oh my God, I can't get this white dude." Struggling. And I was like, but the thing is, the black person in me, honestly, I saw the flaws in the paper in the project.
Okay.
I didn't say nothing, but I helped what I could. But I kept thinking of you, BGS. I kept thinking of Nameless, Dr. Dunbar, T.S. Johnson and stuff. I kept thinking Kendra. I kept thinking of like everybody I hear, Topaz, what she's doing with psychology and everything. And I was like, would he do the same thing if the shoe was on the other foot? Hell no.
Probably not.
Get my minimalistic view of this. And that was it. Like, no, I went straight to the paper. What's on paper that I can help you with? That's it. No creativity. I actually took some things he was trying to develop, and I was like, I drew it out. I was like, well, I could flip this into something else, but I didn't do it. But I was like, I could flip this into something else. Oh shit, okay, and I thought it, but I haven't put it into fruition. I haven't tested it myself, so it's just out there, on paper.
Yeah, one thing you earlier were talking about as far as some of the stuff that you were doing, that was really interesting to me, that you talked about some of your work could help with transplant rejection.
Right.
And I know that's a serious problem in the transplant community, because I have a friend, she just got a kidney transplant, and she's having issues with her kidney because with the rejection. So
yeah.
Well, I know, because I think, if I'm not mistaken, if you have a transplant, you have to constantly be on the immunosuppressant drugs to keep your body from attacking the new organ.
Right.
Yeah, I think that's what happened to my niece. Unfortunately, she got a kidney transplant and eventually passed away not too long after it. I didn't really get the details till I talked to her brother about a couple years ago.
Yeah, I know rejection is a huge problem with with transplants. So I'm saying I said, yeah, we can see you. Was it Norway or is it Sweden? Getting the Nobel Prize if you can solve that one, baby.
Dr. Dunbar showed it a way you can do it. A way you can you can deal with it, and they are doing some trials and some experiments with it. I think with rats, they're doing some things. As far as rejection, transplant, they've been doing it for a while.
Wow.
Using the adenovirus method, where you're transfecting certain genes there into those cells that are forcing those cells to not reject. Because of course, you're going to be on immunosuppressants. The issue with immunosuppressants is you have to constantly take them.
Yeah.
Maybe you can reduce the dose. Like, yeah, you're going to keep taking it, but you don't have to take it at 50 milligrams. Maybe you can take it at 5 milligrams, which saves you money over time.
And plus makes you healthier, because if you're on immunosuppressant drugs, that sets you up for other other diseases and pathogens that could get into you. Yeah.
Yes, increased quality of life. That's like number one when it comes to, like, when you read stuff like these trials that say, well, we're evaluating the safety of this. They're trying to improve quality of life for the patient. That's like really important. And so, like, for me, you're the person to transfection for something you understand, which is transplant rejection. Then and it's effective. It works. Like, okay, the person is not rejecting this new tissue, these new cells that are there. Okay, this is working. Let's keep them on immunosuppressants for, at a low dose for like 6 months and see how they do on it. They're less likely to have to keep coming into the doctor or keep coming to the hospital, where they're putting themselves at more risk for infection. So that's what I was.
You keep this up. We keep this up. Basically, the Chinese are going to do a drop squad on you, baby, and kidnap you, and you wind up waking up in Shanghai someplace, working in the lab.
Working in it with the.
Yeah, you and the bat woman working side by side. Oh, I heard about your work.
She was like, I heard about your work. Oh, I thought she was dead.
No, she just in fact she's alive because the WHO just interviewed her like
Really? She's still living? Okay.
She's still living. You know,
she's still around.
She can't talk much, but she's still living.
Right. Is that the woman who, well, quote unquote, let the virus get out?
No, I think the one that let the virus get out, which was a student, I don't think she's around anymore. I think she was one of the first casualties.
Oh, okay.
Yeah, was nobody seen her since?
Yeah, she's gone, unfortunately. But the Bat Lady, it was her laboratory.
It was her laboratory. Yeah, she's the expert.
Conducted.
Yeah.
Mm-hmm.
Well, welcome, Black Uru.
Yeah, thanks for having me up, Dr.
Sorry about the technical difficulties. I don't know what's going on.
The YouTube, man. It's a YouTube problem.
Yeah, it's a YouTube thing.
Yeah, they're messing with you, doctor. They're on to you. You and Nikole at the same time. You don't know. We can't have this. Too much black excellence at one time. No.
Yeah.
Stop them from speaking. Stop them.
I got it from headquarters.
Mike, where I didn't hear when Dr. Dunbar, you were saying something, and I was like, wait, I thought it was on my end. I missed a couple words. I was like, what is going on over here? I'm looking at the screen, like, what's up? And then I picked up and I was like, okay, I know what he was saying.
Did you see a same black cat walk past your house twice?
No, I didn't.
Oh dear.
Nikole, while listening to you, Nikole, you try my inherent rejection of gynocentric language. As I listen to you, I can't help but think of you and speak of you as being a queen of science, which is obviously how I tend to reject using that reference, but I very much appreciate the generosity of the information that you provide in this environment. It's a rare gift you bestow upon us, and I cannot express enough appreciation for that. I have a question for you, Nikole. Maybe you could provide some insight as well, Dr. Dunbar, on the issue. You were speaking about the difficulty men, or I guess male researchers in particular, in terms of garnering support, financial support for your studies. Can you describe maybe the political environment, or how can lay people assist you, in particular in terms of encouraging and help to direct the political environment to steer resources your way?
Oh, there's lobbyists, because we have, in order to get the funding, you have, of course, for me, it's through a National Institutes of Health and DOD, many other Oak Ridge National Laboratory, like all these different laboratories and stuff. And so these things are approved by Congress. So, anything that's going to be lobbying for STEM or for that type of research, very specific type of research in your state, at the state sponsor universities or hospitals, speaking with those companies that are lobbyists, and those congressmen and congresswomen. That's the main thing. That's how a lot of lay people, in my opinion, a lot for what I do, a lot of them are patients, and they're also lobbyists, because they have begun the process of, they didn't just sit down and say, I'm just going to die. I have cancer. I'm dying. They said, let me get involved and help this process to continue.
So do you have family members who have certain conditions? It may be a good idea to help assist them in terms of lobbying to provide the resources you need.
Also reviewing the grants that we put out. I have to do a technical abstract and I have to do a lay abstract. Lay abstract is normally for patients that are reviewing the grants as well. Lay abstract is very down to earth. It's just speaking to you like basic terms. Not a lot of acronyms going on. I actually spell out the word of the acronym. Technical abstract is just like it looks like a bunch of acronyms, basically like another language. It's just a mess. But that's how we understand what we're studying, because you're selling your grant to a certain population. Normally, for me, that population is prostate cancer survivors, their family members, military. That's who I'm selling it to. So any branch of the military personnel that gets their hand on this, that is within that circle of having access to these studies, they're on the cusp of being funded. Several patients have read our grants, and they give sometimes harsher feedback than the scientists. You have scientists that give feedback, you have patients, who are actually sick right now. Because with prostate cancer, you're not always just laying in the bed somewhere. You can still have a regular life. You're just aging and dying on the inside very slowly. Patients who are survivors, patient family members who are survivors, military personnel that are involved in it. So they're reading this and they're like, I don't understand anything that your grant was saying. This doesn't make any sense. They go through like line 27, page 3. This doesn't, I'm like, oh my God. These are the patients that are telling me, like, you didn't think this out very well. This is not a sound concept. And here's a reference that proves that it's not a sound concept. And they give the reference. I'm like, damn. I'm like,
oh, wow.
Like, what? You got a point. Like, that wasn't
when you have a point, huh?
A lot of the patients are educated too, which is why they're able to get the care that they're getting at the time. A lot of them are very educated. A lot of them are very wealthy, apparently. So, even those who may not be as educated, like a PhD or a master's or something, many of them, they have their businesses. They got all kinds of things going on. We can't forget that a lot of the patients, they were veterans. So they're stupid. They know what they're talking about, and they're like, "Look, I've seen sick people all my life." And they're like, this concept that you have developed is great. Or they'll say, oh, this concept sucks. Like, I just don't like it. And I'm like, okay, they have to give an explanation why and give some type of reference. But that is how the lay people get involved, at least for the projects I apply for.
Yeah. My experience is that, as Nikole said, the largest river of the research dollars come from the NIH and the federal budget, and it depends on how much you can get them to allocate funds in the budget. I recall when I was a student during the Bush 43 administration. I remember some professors discussing that that administration felt like more the onus should be on the private sector as opposed to the government.
Okay.
And some researchers are able to get dollars from the private sector as well. So you have to apply the pressure somewhere higher up on either side.
The issue with private sector, yeah, you get money. You get a lot of money, actually, but Lightbearer probably knows, but you talking about my ghetto renditions. It's actually country rendition, but just to be specific, it's not ghetto.
It's called country grammar. Country grammar.
You can get money from private sector, a lot of money. The issue with private sector is sometimes the work is not acceptable to the scientific academy.
Okay.
And so when you're going to, they look at who funded you, who gave you the money to do this project, who did this, that, and the other. Really? And basically whose palms who greased your palms to get this work done. That's what they want to know.
Now, what's the impact on you professionally in that kind of scenario where there's this perception of
a lot of times? Because you went private sector. If you're working in academia and you get money from the private sector, people in academia are gonna be like, take you behind the private sector. You want to make $1 million a year or $3 million a year underground as your salary, another $3-4 million on your grants, you go to private sector. The issue is like getting your papers to the light of day.
You need to be essentially a mercenary.
Well, I thought you were going to mention.
In other words, they want to file for patents. That's what.
Right.
Yeah. So that's the
profit.
Devices you want to be able to. You're more liquid. You're more. You're globally. You could be globally recognized that way. Private sector. You can float back and forth, do what you want, and you're like a free agent because standard academicians, they're not going to really mess with you, but they're not going to touch you either.
Okay. So you know where I want to go with that, right? The place I always go, ethics.
Yep, I think you were going to do that.
It is a segue right directly into that, and I think it's important because at the end of the day, this kind of determines what people decide to see as profitable in STEM, and it has a kickback to the university because it'll affect what a lot of people end up getting into. And when you can apply political agenda to that by having interest groups that support these ideas because the science will support what they're aiming for. And this is a topic I wanted to talk to you guys about in the future. A lot of the eugenics society have made a lot of their leeway by using backdoors through things like CRISPR and genetic engineering to justify certain sciences and other iterations that are really right there from damn near the Nazi Germany era. A lot of that has been going under different institutions in China, for example. But when you sit down and look at the rhetoric and the justification for a lot of these things, these private interest groups have been able to lever the money for that by using that lobby system.
It depends on the group and it depends on the project. That all of that, you're right. I mean, and it really depends on the lobbying group that you're going with because they have their own agenda, their own justification for why they're lobbying in the first place.
Yeah, a lot of we're just going to call them a lot of the intersectionalist groups, for example, get a lot of that grant money tied up because they're able to, take their political power and leverage it to get that financial backing. A lot of times they'll be the ones behind certain people getting reelected in the office, for example. And so, when it comes down to applying for that money, and, Dr. Nikole Ali brought that up a second ago, you asked the question, it's why certain demographics are getting essentially first dibs on a lot of this stuff, even though there's people just as qualified, if not even more so, that aren't getting to see the light of day for a lot of this stuff, let alone even getting to go to school for some of this stuff.
Mm-hmm. I've seen that firsthand. Especially here in LA, I've seen that, we have a dying population of people who they're getting older. And I know I talk about the veterans a lot, but, you have male, female veterans, obviously. Population that's having so many issues with, homelessness, substance abuse, cancer, diabetes and cancer. Actually, it's diabetes and cancer. That's like number 1 and 2. They kind of flip back and forth. Those two right there. And just lobbying for that. But at the same time, any hospital, LA County, any hospital, any clinic, you will see transgender on there. When it comes to like elderly stuff, it's like all the way at the bottom now. It's like, oh yeah, who cares about you? You want these transgender people, and I'm not saying nothing against them as a demographic, but it's just the way that it is presented, the optics of it, to me, is unethical.
And it's getting into even areas like psychology, where a lot of the terms and baseline definitions in like the American Society for Psychology, they're redefining attributes to masculinity and femininity, for example. And it's really, this is how science gets affected by the politics, and it really seems to stem originally from the universities. And as the funding for that really ends up showing up, I guess Dr. Tia-San Johnson's an excellent person to bring a lot of this stuff up too. This is where all this comes into. Go ahead, BGS.
Okay, well, this is what Dr. Johnson told me. We call it they take the trash out on Tuesday. Okay. In other words, what he says is that especially in these higher level institutions with these privileged students, okay, they get indoctrinated into the university, right, in the school of thought. And when they go out, they don't just go, they don't just start as garbage men and work their way up. Basically, they go into HR, they go into junior CEO positions and stuff like that, or they can be aides to politicians. So they, right from college, they go right into these positions that are influential. And within 5 to 10 years, they move up and they are the people. So that's how come it shows up not only in politics, but also in business, because they've been indoctrinated 10 years earlier in these schools of thought. And it takes about 10 years for it to show back up. But it's not the, not the slight Nikole. It's not the Nikoles, but it's the Nikoles that, that went to Harvard, or the Nikoles that went to Yale, the Nikoles that went to Stanford, okay, that have parents that are already influential, that have, already have money, and they get slotted right in.
Right.
And they carry those schools of thought with them.
Whatever.
And they get maneuvered into choice media attention.
Choice media.
So the public get to learn who they are very early. It's not coincidental who appears on CNN or MSNBC.
But also they come in as a wave, okay, as a wave of people that are influential, and as they move up the ladder, these policies start to see these policies. If they don't change their worldview, their school of thought, when they come in as a wave, you don't notice them. You see it on the university level. 10 years before, right? But after 10 years with these higher level people moving up into the, when they get like 30, 32, 33, where they move up to the higher positions, they can actually implement these policies in business and politics, in education and every place else, because they're not coming in as individuals, coming in as a whole group. Does that make sense?
I see that exactly with the situation that happened in Houston, where you had all of those individuals copping out of law school. And this is kind of how I came across a lot of this stuff personally. A lot of these folks coming out of law school, getting into positions of being heads of police departments, and getting to work with these individuals from the ground up. And on the sorority level, a lot of them get fast-tracked into some of these positions, and they'll end up taking over whole blocks and cities. And it's just like you said, the leapfrog method, where they'll put themselves on, and if you're from the outside in with no network trying to get in, you're not getting anywhere. And even if you do, unless you've got a network that can protect you from the HR level, all it takes is a single incident for you to gain some sort of, potential civil rights issue against you because, you used the wrong pronoun or you got Me Too, and, it's a wrap. And, a lot of people have to walk around keeping their head down and their nose clean with regards to that, while you've got other groups of people who can get away with murder. And so it's why we get a lot of these difficulties in bringing some of these issues to the forefront, which again highlights what Nikole's doing is nothing short of amazing, because even she had to go through DOD and present a motive or a modality of interest, because obviously who's the military consisted of as far as folks that are going to be coming back, getting injured as veterans and whatnot, both physically and psychologically. And then we already know they're already underwater and under heat over what's going on with the VA and a lot of the malpractice issues that's been going on there. So anything that's going to clean up that rep is going to be positive. But that was a good line of thinking. And hopefully more of us kind of get an idea of this bigger picture, because there's so many more technologies and things like this that are essentially being hot potato to other groups of people. And the United States does have an interest of trying to play catch up, especially with regards to China. I think someone mentioned that in the chat. And I think the answer to a lot of this is women in general, and then other groups of people like the Hispanic demographic for numerical reasons, followed by trying to adopt a brain drain policy from other countries, particularly India, China, and believe it or not, a lot of recent portions of Africa.
Yeah, I agree with that.
So you guys can probably look at your current colleagues and see a pattern among that, like already where you're at the level you're at, the PhDs here.
Yeah, there are a lot of things that I would not do because of the ethical standard. We have an ethical standard. It's not the same as the Hippocratic Oath, but we still have a standard to search for the truth. Basically, that's the standard. Above all else, seek the truth. That's a basic standard. in the sciences. And with, even if it hurts you, well, I got the truth, I got what I got, but you don't want to hurt other people. I guess it's like Hippocratic Oath mixed in with that, because you don't want to hurt other people. Seeking the truth, I will kill 100 mice before I hurt one patient. That's just how I am. A lot of people are not like that necessarily. They might do, especially in the psychology field. And my friend, I'm about to go see her later on today. She's in neuroscience and psychology. So a lot of the markers that she looks at UCLA are with, they are markers that would go up and down with different psychotropics, psychotropic drugs. So when she's doing these analyses, she's not doing it in humans. It's all in rats or hamsters. And that's her ethical part of it. I mean, she's probably killed, who knows, thousands of animals trying to do these experiments. She's gotten her funding from different sources, private, some mostly public, but some private as well, to get it done, because they noticed like what Nameless is really focusing on is like the pinpoint accuracy of their attention to detail. Like they noticed one side effect when she did her technique, which was, they had expressed less of these genes that are contributing to depression, but their eyesight got better. You know what I'm saying? So their level of acuity, visual acuity got better. And they were like, oh, yeah. They're less depressed. Okay, yeah, whatever. About this eyesight stuff, they wanted to focus on that. And she was like, well, that's not the primary focus of my project. Like, that's not why y'all gave me the money. We'll give you more money if you focus on the eyesight. Because of the eyesight, you have, it opens up a whole new can of worms. You have diabetes, you have retinal neuropathy, you got macular degeneration, you got angiogenesis, which links you back into cancer, which she didn't want to do. All these different, I just named 4. They had a whole laundry list. I saw the list. I was like, damn. How you gonna do all this with one grant? She's like, I guess they're gonna have to give you multiple grants. I was like, okay, do your thing.
Yeah, because it translates back into money.
Right. And the ethics there was not how statistically significant her data was at reducing the depression of these mice or these rats that she was using. They didn't care about that. I was like, this is something you can translate directly into humans. They were like, yeah, we got plenty of drugs on the market. Yeah, whatever. About this, the eyesight stuff, that's what we really need to look at. They already had, I told her about this, and she heard it before I did. Because when it comes to neurological studies, she gets it before I do. And then I pass it down to y'all. I give it to y'all. I'm like, okay, y'all go ahead and take these little notes. Take these notes. So when I have my videos, it's like I'm passing notes to, these are a lot of other people I know, they're the ones that like, hey, she gets the articles like 3 days after it's published, because that's neurology. So when I'm telling you all about that particular project where they gave the mice infrared vision, you had to teach me the concept before I could teach it to y'all.
Okay, so this is in other words, cutting edge stuff.
Yes. And she was like, yo, this is some real stuff right here. This is real. I was like, if you don't take your drunk high, I don't know what's wrong with you. Like, you've been in the lab too long, drinking too much coffee. I don't know what's going on. And she was like, I'm not drunk. I'm not high. I'm just telling you, this is some real stuff. They're going to have this all over the place. And I looked at it. I said, oh, this is Black Mirror episode. I was like, oh, dang. And she said, we had never heard of Black Mirror. Because, her job is Black Mirror. Like, a lot of stuff
exactly. Exactly. But you are spooked to sit by the door. Yeah.
And I think, she is as well. And I mean, she does the, she does a lot of work that I don't want to do. And I, I don't want to do that. But, she does the,
that's how we get stuff done.
Injections. But I don't want to do it. I'll just rather train somebody, pay to train somebody to do it. I don't want to do it. Like, she can do a lot of eye and brain techniques. I can do more vascular surgery techniques, which is below the neck. I don't like working above the neck with the head and everything. I don't like doing nothing there. Carotid artery type stuff and brain surgery and cathodes and injections in the eye and all this stuff. I ain't about to do all that. No.
You all are sedating the animals, of course, right? Or
when you do an intraorbital injection, they're not sedated.
Okay.
They're just slightly kind of out of it, gassed a little bit, but they're not, or they give anesthesia a little bit. They're not completely sedated. Their eyes need to be, open. You need to be able to tell if there's like a hemorrhage or something, if you've punctured something wrong, because you want them to be able to see after you give them that injection. It's hard to do that. Blood pressure is so low, you won't be able to see the effects if you made a mistake until they wake up, because their eyes don't close, right? Their eyes stay open all the time. And injection is yeah, they're pretty much awake. They can't blink, so that's the good thing. But she does a lot of things that I don't want to do, and then vice versa for me. She's like, I don't know how you do this. Like, why you, you're gonna just inject a whole lot of tumors into the prostate. Why are you doing this? I was like, because I need to kill this mouse to figure out what's going on. And she's like, and I'm like, well, she's like, why are you doing that? I'm like, I'm taking patient tissue, taking a little piece of it, putting it into the mouse, waiting for it to grow. What's the problem? She's like, you're weird. You're Frankenstein. I'm like, I'm Frankenstein? With a chip on his head, running around with hair sticking out of his head. But I'm Frankenstein. Really though? Really? We're going back and forth like this. This is like 5 years been going back and forth. And I'm like, I'm telling her, I'm like, really though? I said, you can't tell me nothing about no Frankenstein when you got mice and rats in there. And they have wires literally hanging from the ceiling connected to their head with chips on it, and they're running around being tested and monitored and stuff. Gee whiz. Okay. That is that's old school, but that's what the chips are not that big now. Now they're like, they're not even, they're smaller than a stamp. They're still small. Ooh. The chips they use. Like you can just implant it and then close the skull up and then go. The old ones, they used to, they were hanging out. It's just sitting on top of the head, on top of the brain. So I was like, you telling me about some, I said, at least when I'm done with my mice, I sew them up and put them back in the cage. You can't even sew yours up. They're just out. Like running around. And she was like, yeah, you got a point. Like imagine 20 mice are rats because they have bigger heads, but you got 20 rats and they're running around. And they're got these huge chips that are about maybe 2 times the size of a stamp on their head. And they're lighting up and stuff. And, you're doing different experiments as far as drugs, giving them different drugs, seeing what they do, seeing how they interact with their babies. And you're tracking their movement. They're monitoring their neurotransmitter productions, all that kind of stuff. Because that's what you can't blame anybody but yourself, because you wrote this grant. This was your idea in your head. Mm-hmm. Come to fruition. So you're weird. You're a weirdo. Get over it. You're a weirdo. That's like, for everybody who writes grants out there, I know Rice has written grants and stuff. She had a discussion with you about that once, BGS. Mm-hmm. And she and I, we have already accepted, we're weirdos, whatever.
I don't know. I don't think you're a weirdo. I think, you're not
from the outside world. Okay. It's you look at the concept of how I developed the concept. Well, you don't want to see that room, but it's just like notes to paper. It's like a beautiful mind or something. Rainmaker, it's a bunch of stuff like, oh, I could do this and I could do this and I could do, put this together and put that together. Bam, there it is. It'll work.
Every brilliant person I've ever known in my life operates the same way.
Mm-hmm. Yeah.
Everyone I've ever known.
It's all about the ideas.
Yeah.
When they come randomly. They come at inopportune times. It's nothing neat. There's very few things that are linear about genius. Very few.
Oh, Snuggles said who makes the chips. I don't know. That's a good question. I know she programmed them. She wrote the program for the chips, but I don't know who made them and put the program on it, because she does programming and stuff on the side, and she programmed.
So Elon Musk hasn't snatched her up yet?
No, she just living her life. But I mean, she's like, I'm I'm like, that's how I feel too. Like, it's just like, okay, well, there's like tons of other people that do way more, way cooler stuff. And we just trying to make ends meet and just, make the money and do our job. Like, that's really it. Come with more ideas. Just have more ideas. Like everybody else, we just regular people, just paying bills and going to work. And sometimes your ideas are too cool for school. And you got get kicked out. Sometimes they're too cool. You blow something up. That happened, but you just have to really. It's the environment. Really, your environment is putting you in a particular mindset where I could do anything, and you just write it out. About 90% of it is written out and literally on pen and paper of how it's going to work. What are the cells doing? How are they talking? What are these genes doing? How are they talking to each other? Things like that. And once you write out all the steps of what you need to do, at this point, for me, I can just give the protocol to somebody to just run the experiment. And I already have an idea that this is going to work in my favor. It's going to answer this question. Like Dr. Dunbar said earlier, it's all about your question. What are you asking? Are you asking, does this drug make the tumor growth go down? Are you asking, does this drug make this tumor gene go down?
Yeah. You know what? I heard I can't remember his name, but he was a very famous engineer, and what he said is it said the answer is not in the solution. The answer is in asking the right question.
Right.
Yeah, that's where the idea comes from. Absolutely,
that's it right there. Because then it forms all the research you have to do to find out. Well, I'm not the first one to think of this. Who else thought of this? And then you look up. That's your references, your introduction right there. And then you're like, oh, wait a minute, they skipped something, and that's the wild idea that you have.
Yeah.
The wild idea.
What did they miss? Yeah, what did they miss?
Yeah, they missed something. Sometimes they'll just tell you, we've missed this and that. And you don't see anything else from them that talks about it. You're like, oh, I'm gonna do that.
Or like which is a portion that I love. One of my favorite quotes is when Tony Stark's father was talking to him, and he said a lot of the problems that I can't solve because the solutions haven't come yet, or the infrastructure is not ready for it yet. And it said I can't solve it, but you can, okay? Because we have advanced enough, and the tools that you need to actually solve this problem will be there. And I think sometimes that happens. Like you see a very old problem that's been sitting around, but it's not time for the environment's not ready for that answer to actually come yet. And I think a lot of times that happens. Okay, that person couldn't solve it because the tools weren't there to solve it yet. But thing is, you take somebody like Nikole says, okay, I know what she was trying to do, and I can use this, and this, and this, and have a break.
Speaking of, I mean to cut you off, Master Teacher.
No, go ahead.
Well, speaking of technology, Snuggle says, what are the tech companies that coincide with the work you all do in the lab, if you don't mind asking? I would say that depends on the nature of the work. Because specific companies make specific things. Agilent, I think, does a lot of the detection, like the HPLCs and the mass specs, and Innova will make your orbital shakers, and it just depends. You want to say a few, Nikole?
Biorad makes a lot of equipment that we use. Applied Biosystems makes a lot of the real-time. Well, they did for a long time real-time PCR machines, but now it's more. They're becoming more multiplexed. And Agilent, definitely, and I mean, because there's like tiers of tech companies now. They're like all forming a huge, conglomerate with each other. It's like a Walmart. They're turning into Walmart in a way.
Yeah, and we're not even including the software that
software
that comes with it.
Yeah.
Everything has a driver and
and the back end. Like IBM is huge in the medical field because of the AIs. You know.
Yeah, some people ask that question, like on Sole Provider's channel. They ask that question because they want to know what they can invest in.
Mm-hmm.
And yeah, a lot of these companies doing well. The problem is, like I said, they're merging so much. Like Thermo Scientific, Fisher, Millipore. That's all one thing. They used to be like 3 different things.
Yeah, the biotech is following the way of what happened in computer tech like 20 years ago.
Mm-hmm.
We have all these little bitty companies that are forming with great ideas, but they don't have the money or the other tools to go along with it, and they get bought up by bigger companies, or they merge until we have these big conglomerates like Microsoft. Microsoft was just a little bitty software company until IBM came in and funded them. You know, and then they started buying up bits and pieces of other, other good ideas and incorporated it into what we call Windows. Yeah, they never stop. You know, you look at even what we're using now, StreamYards. StreamYards was just bought.
By who?
I forget the name of the company, but they just merged with a. In fact, you go back on StreamYards, they'll tell you who they were merged with, another streaming company, because StreamYards had stuff. Well, basically what they were saying is that they were more like Zoom, where they're more like conference, right? But they had, a lot of people that did conferencing use StreamYards. So basically, it said, okay, why don't we just incorporate what StreamYards had, their tools, with us? So they went in and I think they paid them $35 million to buy them.
Okay.
So yeah. So it happens all the time.
I could tell you, setting up server to make sure that stuff properly run is one headache.
Yeah, I think because I think the guy that's doing OBS Ninja, he's following that same line. So but most companies like that, they come up with these great ideas and do the work enough to where they can be bought out and cash out. Yeah, so that's
you have to, because like just to run YouTube. When I hear black people say, I'm gonna start my own YouTube, I'm like, good luck.
Well, yeah. Even though
doing all that bandwidth and stuff, I don't want that headache.
Yeah, that's true. That's true. You got to have good engineers, which is why when we do get black providers like that, we need to go ahead, and even if we don't use it very much, go ahead and give them the $10 a month. That's why really StreamYards. That's like StreamYards developed so fast. They started off free, but it was good, and I said very early on, hey, if you wanted to get better, you're going to have to give them their $20 a month so they can get better, because the more money they have, the better, the more people they can hire, and the better your software can be. Just like say, like Nikole should be, in our sector, Nikole should be a multimillionaire. And I know Jason would love to hear that.
Yeah, he was like,
multi-millions? Yeah, okay.
Yeah.
So Nikole, we have an interesting question.
I see on the screen.
I'm sorry.
I don't think Monsanto ain't got nothing. Well, I wouldn't say.
Well, yeah, let me read this, because I think this kind of delves into both our areas. With everything you have been studying, do you think the company Monsanto has anything to do with the change or this rapid growth when it comes to cancer? So from my end, Monsanto is a chemical company, and they were making a very high profile herbicide. I'm not sure I should say it here because I kind of worked on this, but they also that herbicide is used with GMO crops,
and especially soybeans.
Soybeans and corn.
Yeah. I remember back when I was trading. Yeah, that was a big thing back in the mid '90s. Monsanto. Yeah,
so there was a huge hysteria over GMO. So Nikole, have you seen anything about is GMO dangerous in terms of cancer? Or
yeah, absolutely. Yeah, you can look up. There's documentaries on it where Monsanto they actually hired people. The FDA. I think it was FDA. If I'm not mistaken. USDA, FDA actually hired people who were former Monsanto executives and employees and things to regulate Monsanto. You have Monsanto regulating themselves, like at one point. But the tumors, I'll never forget the tumors on the mice. The thyroid tumors, they're just hanging off of the side of their necks, where they can't. I'm sorry, rats. Excuse me. Where they can't even barely walk. Some of them hanging on the mammary fat pad, causing breast cancer in the female. rats where it's just they can barely walk because the tumor is lifting their forelimbs up.
Really? Oh my goodness.
Oh wow.
Sit there. They just sit around. It's hard to eat. They have to, manually feed them and give them water to drink and things like that. It was devastating, and to see the animals feel like that. And I said, wait, that happened in a very short period of time. For humans, it's not expected to have any results for like 30 years or so. An issue with it was a black man who was a groundskeeper. I think it was last year or something, or a year before, and he had that huge lawsuit with Monsanto for Roundup, chemical spray, the herbicide spray. And the issue with that was like it had occurred so quickly. It wasn't 40 years, 30 years.
Yeah.
So yeah, there is there's definitely a effect there that's inducing cancer.
Yeah, because back in the day, because I was talking to the farmers and with the Roundup Ready beans, the way they could grow, they could actually just literally pour these herbicides onto the crops, and it would kill the weeds and the insects, but it wouldn't damage the soybeans, the soybean crop. So I think back in the 90s, a lot of farmers are overspraying.
Yeah, it'll get worse because people are getting older, living longer now. See, if we had Monsanto back in the 30s or 40s, you wouldn't have noticed it as much. People didn't live as long.
Yeah.
Now people are living longer. Like I said earlier, we were talking about aging. You live long enough, regardless of Monsanto or anything. I mean, you have people in this country that will never be exposed to Monsanto or Monsanto products, but they still get cancer in those areas.
Just from the mutations that naturally occur.
It naturally will occur after you age so many times, after your cells have so many cycles.
Yeah, I'm serving on the cancer group in my current division, and yes, there are just some. Yeah, you start to see increased tumors as the rodents age, even if you don't treat them with a molecule of any kind.
Right. We just treated them with saline control group after these were mice that were engineered to age faster. So, they're only supposed to live like a year. These mice were like genetically, they were like 3 years old, but they were 6 months old. And they had tumors and stuff. I was like, we had to keep starting over because, start at a younger and younger age, like before puberty, because they kept getting tumors and it were no treatment, just saline treatment. Like I was like, I can't figure this out. Like we were trying to induce they were getting ovarian tumors because it was female rodents we were using, and we were trying to cause an inflammatory response in the ovarian within the ovary, so within the uterus, within all of that. We were trying to cause like an inflammatory response using bleach.
Bleach?
Yeah, like bleach, different chemical. That's a chemical, and it causes an inflammatory response, and it flushes right out. Right.
Okay. So there was some precedent for
yeah, I was gonna say you're gonna start.
I'm not gonna say it. I'm not gonna say it.
Inflammatory response faster though. That was the issue.
Give it a little S curl.
Yeah, it was. Yeah, that was hard to do. I was like, why the hell? But we had to do that because we were looking at all the chemicals that cause that inflammasome, because that is the body sees it as obviously toxic, very alkaline. And it's like, oh, wait, we have to return the cells to homeostasis. And so you just like flush the whole system. The ones that were older, they already had the inflammasome. They had like a cytokine storm within the uterus. That was the problem. And I was like, damn it. We could not get that right for a while. Finally got it right and did the right thing, with using younger and younger and younger mice. And then we had rats because mice are cheaper. So then we tried it in the rats. It worked. And I was like, okay, that's cool.
Yeah.
One question.
Go ahead, AB. No, go ahead, AB.
Just out of curiosity, what's the difference between experimenting on a mouse and a mice and as rather than a rat?
They're smaller and cheaper. They have more. They have excuse me, more pups in the litter usually than rats do. Rats are bigger. They're like 4 times bigger.
Yeah.
Sometimes than a mouse, and they bite really hard. They can grab really hard. They grab. They have little paws and stuff.
Okay.
Big, and they're very territorial. The males are very aggressive.
Okay.
And more space, so they cost more money. That's why. So usually I start in mice and then I transition into rats.
Okay.
So since Nikole and I were talking about aging earlier, I can tell you that for cancer bioassays in mice and rats, the mouse study, this is for a pesticide, it's usually 78 weeks, and the rat is 104 weeks, because that's essentially the lifetime, right? Because Nikole, you're working more with mice. So the 78 weeks, that's a lifetime, right, for a mouse?
Yeah, that's a long time.
Yeah, and then 2 years is a lifetime for a rat.
Rats 2 years. I usually sack mice or euthanize mice around no more than 20 weeks.
Really?
Wow.
Oh wow.
Okay.
Like we started experiment like they're maybe 8 to 10 weeks old.
Okay.
Another 10 weeks, you gone. Like be holding on.
That's not nice.
I don't care. Like my mom tells me that. That's how long did you have it?
Yeah, that's me. That's mean.
I let her read it, and she's like, "Well, how long did you get them?" I said, "Well, we started. These were 8 weeks, and when'd you sack them?" I was like, "I sacked them at 15 weeks. 15 weeks. Oh, poor little fella." I'm like, yeah, I don't care about it. I don't care about it. I was like, man, you gotta go sometime. I mean, they that's all it is.
It's a soldier's life, huh?
Yeah, yeah. Hey, I'd rather get rid of them than hurt any human.
So Nikole, I was about to ask you if the rats fight the way the mice do, but you're not likely to put as many rats in a cage, as you will with mice.
No.
Yeah.
No.
You said the males are very territorial.
Be ridiculous or be overwhelming. That's why the cages for the rats are like maybe 2 times bigger, maybe 2 or 3 times bigger. Depends on if you have males in there or females. If you have females, they're about 3 times bigger than a mouse cage because of their pups.
Okay.
And they want to have their own little, their own little apartment in there. The females, they start when they're pregnant. They are, they're nesting, getting their little nest together and stuff. And they don't want each other touching their stuff. Like, don't touch my stuff. Going back and forth, fighting. Why'd you take my pillows? They're literally a fight. So we separate them. You can have, I think, you could put 2 males in a cage, rats, and they'll be okay. It's like for some reason, like the 3 or 4 male rats in a cage, that the waste is ridiculous, and they will fight. They run out of space. We can do 2 female rats and 1 male rat. We cannot do 2 male rats and 1 female rat.
Is that where you got that concept where you talked about the male rat couldn't even turn around on another male rat because he'll get his
all the time.
Bite the tail,
bite the tail, bite the nose off.
Well, sound like the manosphere.
Yeah, I remember you. It's like wow.
Pretty much. I hate to say that, but
you bring in one female and a panel of men, and all hell breaks loose.
You literally see like their shoulders go down.
Ironically, I saw that this morning too.
They literally get like you can just see their shoulders go down. They're like, oh, that's a female in here. Okay, they go and they smell her like, oh, okay, it's a girl. Okay, hey, what's up? They talk to her and stuff. She's like, yeah, whatever, and she's just walking around doing her thing. She's nesting automatically. She just starts doing that. Like when you put a female in the male cage, in it, It's a mess. It's like it's scattered everywhere.
Yeah, start leaving a pair of shoes here, a spare stockings there, you know.
Yeah.
Wash out the underwear in the sink, and you know.
Yeah.
Get a little toothbrush, you know. So yeah, just start
I sat and watched them. I was like, okay, while I'm weaning the mice, or I'm sitting here, I'm trying to determine the sex of the mice, like, okay, male, female, separating in them. I'm looking at them while they're interacting, you know, 2 males and one female. I'm like, well, that ain't gonna work. Like literally within minutes, it's like, you know, it's a mess. Like they greet each other and they're like, yeah. And she's like over there cleaning up. And then, you know, while she's cleaning up, making her little nest for herself, they're over there like, you know, fighting each other. Well, I saw you looking at her. I saw you looking at her.
Oh, gee whiz.
You know, fighting just within minutes. It's like they're just fighting all over the place. And I'm like, you can literally hear them like boom, like running around in circles, running, making a little track. He's just sitting there and people like looking at them. Like,
that is hilarious. That is hilarious.
They're very acrobatic.
Oh yeah.
They're climbing on the bars upside down, and
oh wow,
very acrobatic.
I've seen them do that. They'll climb upside down and like a little acrobat, hang upside down while they're fighting. The way they're fighting and stuff.
The other fascinating thing,
like they're in the Matrix.
The other fascinating thing is how they all sleep in a ball. So if you walk in and they're all kind of like you like this one big black, whichever string you're using is this one big ball of them all sleeping together in a corner.
Yeah, they do that just to retain heat because it's usually cold in the facility, mouse facility.
Oh,
yeah, wow. They have padding under them, and they're just like little cows. They have like the padding under them. The women are organizing the padding, and you know where the pups will sleep is in the middle.
Yeah.
And just for protection, you know, that's just an innate thing for protection. Put the pups in the middle and they sleep around them if they have pups there. So they do sleep in a little. I've gone in there and I was like, oh, the first time I saw it, I was like, what? I had black 6 mice, like big black ball of fur. I was like, what is all this? Because all their heads were down and their little tails were curled around and they were sleeping. I was like, oh, look at that. Yeah.
Let me inject you with this. Wake up.
And they'll be sleeping all together, a little family, and they'll get up and just be fighting. I'm like, ugh. I know y'all black folks.
Yeah, sound like the black community.
Yeah, the black community's
I know y'all black folks because y'all after you go back up fighting, like you know, when with the food and fight over the food and no, I need some water first. Get away because they wash their faces and stuff, you know. And then they get a little water and they like rubbing their hand, like, you know, wash their little face and, you know, clean off and stuff. Like, no, man, get out of the way, move, like
mouse bath.
Oh boy,
girl.
So Nikole, I think you answered this one, but I'm going to add that most of the reagents, well, we know the technology that's purchased, but most of the reagents, like the buffers, even just the buffer, that's purchased from a company like Sigma as well. And I think this gets to the cost. There's a significant cost to conducting the experiments.
Yeah, you have to learn how to mix up the chemicals yourself. That way you can just buy the as long as you have access to nanopure water, you just buy the chemicals dry and crystallized, or if you have certain reagents that are lyophilized, you just need water or DMSO or PBS or something.
So what is I know purified water or distilled water. What is nanopure water?
The nanopure water is like what is it called? We call it millicube. It's like it's a sensor. You have a sensor that purifies it. There's the standard filters and everything there. To me, it's just monitored much better than the regular standard filter that you get.
Okay.
We filter like 3 times. And even with that, I don't trust it. So I still, even though it has the readings and it says, you know, 98% pure, 99% pure, which is very high. I still have to take that water and autoclave it for certain processes.
Okay, because back, because I'm sure Dr. Dunbar knows, is old school. You have to distill water, so you have to boil a lot of water and distill it down.
Right.
To get the water to pure water to actually mix whatever reagents you're gonna mix with it, whatever test you're gonna run. So that's what I was wondering. Is it? I guess it's not as pure. It's not as good as distilled water, but it's closer.
No, it's better than distilled water because
oh, really? Okay.
Distilled water is just like you can buy that, straight from the company, but they don't necessarily have to tell you if it's been how many times it's been distilled or anything like that. It's just whatever they have on hand. And so even if I order distilled water from like Sigma or somewhere, and I get that, I still have to pour that out and autoclave that. I have to pour that out of the plastic container, throw the plastic container away, pour it into a glass container.
Yeah, why would we put distilled water in a plastic container?
Because they think that it's not going to be used in that container very long.
Yeah.
But we store everything in the that thick Corning heavy glass.
Heavy
glassware. That's what we store it in. Put a piece of autoclave tape on it and autoclave it.
Yeah, that's what the first thing I learned, and back when I was first in high school in Chem Tech, before I had the chemistry classes, is that all this stuff has got to get stored in glass because plastic will adulterate it.
Of course, yeah, it'll leach into the plastic,
into the water.
Yeah, and they go back and forth because depending on how hot it gets, the can occur both ways. Mostly like you said, mostly with the plastic. Leaching into the water, that affects your readings, especially if you're doing something like HPLC. What is it called? The liquid chromatography. You're doing something like real-time PCR. If you're doing anything with RNA, it's got to be like really super, super pure nuclear-free water. Once you take the water out of the millicube or nanopure water system out of the spout and put it into a glass jug, just it leaving out the spout, it's contaminated.
Because of the air.
It's pure, but it's still a little contaminated. It has nucleus from the water, from the air, from the
air outside. It was say anything you can smell gives off gas.
Any aromatic out there, you have like all that is in, it's going into a 2-liter glass jug, bottle with a cap on it that you're going to autoclave, and you're like, okay, I need this autoclave water not only for doing RNA analysis, because I can go through like 10 mils a month for RNA analysis, which is nothing. But you also need the 2 liters of autoclave water for the incubators to keep the cells, in a humidified chamber. You pour water in the bottom of the incubator to keep keep it humid in there so they stay alive. If it's too dry, they dry up, everything evaporates, they die. So you need it for that. You need it for injections. Sometimes certain drugs ask for you to inject a mouse with saline, like 0.9% saline. Others, which is just salt water. Others ask our drug needs to be in water, ultra pure water. Like, okay, I can do that, whatever.
Just out of curiosity.
Water.
Just out of curiosity, what is saline?
Salt water.
Just salt water. NaCl water.
Okay.
Different percentages.
Different percentage. Yeah.
Okay.
Well, hey everyone, we are at 3 hours.
Okay. Yeah.
All right.
So I should probably shut things down, go spend some time with family.
Okay.
Yeah.
All right. All right. Any candy grams?
No. No candy grams.
Is Dr. Goodbar's not working tonight?
No, I don't think so. Nikole, it changed to Dr. Goodbar somewhere.
Dr. Goodbar has swag. It's like Dr. Dunbar. Dr. Dunbar is was it Dr. Jekyll, and Dr. Goodbar is Mr. Hyde. So when he takes his visits, knock knock, who's that? Dr. Goodbar.
Ironically, that was my father's favorite candy bar, Mr. Goodbar.
Mr. Goodbar. Oh yeah, remember the movie Searching for Mr. Goodbar?
No, I don't remember that.
I don't remember.
Or no, Looking for Mr. Goodbar. Yeah, it's back in the 80s. Yeah, go look it up.
I'll look it up.
Look it up.
That's one of my favorite candy bars too.
Well, yeah, we had a different meaning.
Yeah, okay. That's not surprising, but okay.
Oh yeah, it was a chick. Back then the independent women were looking for Mr. Goodbar.
Really?
Oh,
Okay.
Yeah, searching for Mr. Goodbar. So Dr. Goodbar is
so Mr. Goodbar was the first simp?
No. Mr. Goodbar was the alpha.
Oh, okay.
In other words, that's what Mr. Good sex. So she was looking for Mr. Goodbar.
Okay.
And so
why they had her play that though? Wow.
I forget who the chick was that played it, but this is back in the 80s. It was Diane Keaton, yeah, and she was obsessed, and I think she found him, but I think he killed her.
Ooh,
yeah. So she found him. She found her Goodbar, but that was the last one she had. So
what you trying? Moral of the story is,
all good bars ain't good bars. Okay.
Yeah.
At some point, I turned into a high value man. No. Where's Charles? I'm a multifaceted man.
Yeah, multifaceted.
Oh, so now you're a Jedi in the streets and a Sith in the sheets. Oh.
Yeah. There you go.
All right, moving on up.
Before we shut down Snuggles, I'm going to answer this by saying that if the plant forms something toxic and you ingest it, it can be harmful to you.
Yeah, or you could be allergic to certain plants.
Poison ivy, yeah.
Well, you even like say somebody that has like a nut allergy. Okay, won't harm anybody else, but your liver doesn't process it well, so it can kill you. Turn you, basically, you go into shock and your throat closes up.
Ooh,
yeah. Milk. What is it? Milk, nuts,
other kinds of food, avocados, wheat bread, gluten.
Yeah.
Yeah, I just started to try almond milk, just to
Oh, it's so good.
Yeah,
like it.
Yeah, I don't taste the difference. Not really.
Oh, I can taste the difference.
Almond milk tastes like metal to me. When I went back and I had a and I knew it because I knew the lady at Dunkin' Donuts. I know she did not put my almond milk in there. When I tasted it, I was like, "What the hell? I was like, "What is this? This tastes like metal." Did you look in here? Like, oh yeah, my bad. We didn't have almond milk. I was like, you trying to kill me?
I'm glad you said that because when I look back, usually when you get milk or something like that from a place like that, it does taste like metal sometimes. Like you can taste the metal that was in there or something.
Yeah, I think it's the it's not the container. I don't think it's the container it's in. It's just all the processing of the milk and everything. I think that's what it is. It didn't taste like that when I was younger. I mean, we could get fresh milk from like the dairy cow people, down the road, and it was fine. Well, it wasn't really pasteurized, but it was fine. We were good with that.
It's what they feed the like organic milk. All the cows are grass fed. And that makes a difference. You guys are in this field. Whatever you feed the cow, that's what's going to come out in the milk.
True.
So it's a difference between feeding the cow grass or feeding the cow enhanced soybean meal or something like that. All the kind of stuff to fatten the cow up or give the cow protein or something like that. So whatever you feed the cow, that's what's going to come out in the milk.
Right.
Yeah. So if you get milk that comes from like a factory farm where the cows are basically, in the stalls and they're feeding them and they're getting milk out on the other side rather than going out to the field and ranging and eating natural grass. There's going to be a different taste.
Yep. Well, I'm gonna get up out of here, Dr. Dunbar. Thank you for having me, sir.
Nikole, thank you for coming through.
Yeah, no problem. I'll talk to y'all soon. Y'all have a safe day and try to have some fun while y'all stay on a Saturday. Try to have some fun today. But yeah, I'm gonna holler at y'all later.
All right.
Later.
All right.
Peace.
Thank you. And I appreciate you guys for being here, man. You guys don't know.
All right. I appreciate the support, BGS. All right, everyone, we're going to go ahead and shut it on down. Thank you for coming through, and thank you to Darkness for the Cash App. So I will, since the J&J vaccine was approved, I'll probably create some content on that. And Rael, oh yeah, he's laughing at my high value man joke. So yeah, look out for content on the vaccine, and there's some other things that I want to upload on this channel as well. And obviously I want to have Nikole come back again in the future, and Dr. Vernon Morris, who was there in the chat. But as for now, we've been on for 3 hours, and I'm going to shut it on down. So thank you to everyone who came through. Thank you to everyone who donated. And if you're watching on the playback, please like and subscribe. And everyone, be safe out there. Enjoy the rest of your weekend. I'll see you on the YouTube streets. And as always, remember, everyone, that your attitude determines your altitude. Take care, and I'll talk to you the next time. Bye-bye.